This conference will now be recorded.
Well, since I hope everyone is here and we have a lot to cover, we really should begin today's webinar. I really appreciate you all for attending today's virtual conference. A very good morning, good afternoon, or good evening to one and all present here virtually with me today. Let me introduce myself. My name is Clara McBroom, Conference Secretariat at Coalesce Research Group LLC, and I will be available for any of your queries or questions throughout the webinar to help you out technically and organize the webinar successfully. For the next 5 minutes, please allow me to give a brief description about our organization, Coalesce Research Group, and also the further instructions. Coalesce Research Group LLC is very delighted to welcome you all, the speakers, supporting associations, students, and delegates present here virtually with us today. With the aim of the scientific excellence by facilitation, conversation, sharing knowledge, Coalesce Research Group follows through the objective of setting standards in delivering excellence in the research among the researchers and industries, promulgating the research through conducting events and online webinars from various fields of science, especially from the stream of medicine, science, engineering, and technology. With a short duration, we have succeeded in creating a platform for developing a strong professional network with the people across the world from their respective fields that they haven't connected in a while. As a global scientific event organizer, we plan and carry out international conferences, meetings, seminars, symposium, and workshops, providing the best delegate experience, excellence, networking opportunities, conversation leading to innovations, learning new opportunities, lasting work relationship, and professionalization with the professional around the global network. Just recently, we have also coined up our own platform for open access peer-reviewed scholarly literature where all the content is freely available without any charge to the readers. We aim to share the new recent advances through scientific publications, which will help in minimizing the borders and contributing the use of research works of eminent authors, scientists, researchers, research scholars, students, etc., around a broad spectrum of medical, clinical, pharma, and engineering scientific disciplines and its subspecialties to promote the ideas which will guide the development and facilitate with the use of scientific information and ensure its quality and also will be connecting link between researchers of scientific community. We are committed in ensuring that our journals are disseminated worldwide so that the researchers are able to access our content wherever and whenever they need it. As being very new to journals, we have coined up some interesting journals and have also formed the editorial board and we will be very glad if you have any of our today's attendees, the speakers, students, or delegates would extend our support by checking the journals and will be interested in publishing an inaugural issue at any of the journals according to your area of expertise. We will also send an email with the details about the journals after the webinar. Do take your time to check the webinar. Finally, I would like to thank the attendees and participants of today's virtual conference, online international conference on pharmaceutics and novel drug delivery systems, and hope you enjoy the webinar and gain knowledge from the sessions today. We also hope your participation will contribute to new research findings and that will lead to new collaborations and research initiatives. Before we start with the sessions, I guess some of you will be new to GoToMeetings app. Let me just explain you the important tools required for the presentation. On the GoToMeetings app or a web page, you can find four icons at the bottom center: mic, camera, screen, and end. Mic button, click on that to mute or unmute yourself. I request you all to stay muted while our speakers are presenting their research work so that it doesn't cause any disturbance to them. Camera button. You can use this button to share or unshare the web camera. If you have the option to share the web camera, you can do it and it's completely your choice. Screen button. You can use this button to share your screen. This is the main part. During your presentation, after I call your name from my side, I will be making you as a presenter and will give the permission to share the screen. Then only you will be able to share your screen. So once I make you the presenter, you can click on the screen button and you will find a pop-up window to share your screen. End button is the fourth icon where you can use this to leave the session. On the top center, you can use this option to change your display settings. At the top right corner, you will have three icons: people, chat, and settings. In the chat option, you can chat with me or any other participants. For, for the settings, you can use this button to check for the settings of your speakers and microphone. So as mentioned, kindly remain muted during the presentation so that the presenter does not get disturbed. And for any questions, you can ask the questions or you can send your questions in the message box too. Well, let's not take much of your time. Finally, I would like to request you all to please give us a brief introduction about yourself before starting the session so that we can know about you virtually. And now let's move into our session. The first presenter is Dr. Imran Salim from Liverpool, John Morris University in Liverpool, United Kingdom. Hello, Dr. Imran. Are you ready with your presentation?
Yeah, I'll give it a go now.
Okay, Dr. Imran, now I'm making you as a presenter. Kindly click on the screen button to share your screen.
Um,
I have made you as a presenter.
um, right. I'm not able to share my screen at the moment. So what did you say? You pressed the screen button on the bottom?
Yes, in the bottom of your GoToMeetings app, click on the screen button. The mic camera screen, click on the screen button. You will get a pop-up window. Select your PPT in that.
Access. Accessible feature. Allow access. Okay, I think I need to restart the thing because it's.
Okay, did you open the GoToMeetings app?
Yeah, but I think on my computer it's got a firewall thing.
Okay, or you can leave the session and rejoin again. Maybe a technical glitch had happened to the computer.
Yeah, okay. If you okay, let me just restart the thing.
Hello, Dr. Imran. I have made you as a presenter.
Yeah, sorry, I'm having problems. It's not allowing me to click on the screen.
I have made you as a presenter. Have you got any pop-up on your window?
Yeah, it's my computer's blocking me entering. So I'm just trying to see.
I think you need to give a permission from your computer to share your screen.
Yeah, it's not giving me permission. If you wanted, if somebody else wants to jump in while I see if I can.
Okay, can you send your PPT to the program manager? I will be displaying from our end so that you can give your presentation. We'll be displaying from our end and you can give your presentation.
Uh, okay, if I just send it to that email then.
Okay.
1 second. Just gotta find a thing. Yeah, sent that to the email. Have you received the email?
No.
Hello, Dr. Imran. We have
Yeah,
received your PPT. We'll be sharing from our end.
okay,
Uh,
that's
can you please give us a couple of minutes? Hello, Dr. Imran. Are you able to see the screen?
Yeah, okay. Shall I start?
Okay, you can start your presentation. Kindly mention to change the slide when you have needed.
Yeah,
Okay,
okay.
kindly mention the next slide when to change the slide.
Okay, get myself ready. Okay, so I'll just switch on the camera. Okay, sorry about that, everyone. So good morning. So I would like to thank the organizers for the invitation to present my work at this conference. So my name is Professor Imran Salim. So I'm Professor of Nanomedicine here at Liverpool John Moores University in England. So today what I would like to do is present some of the work that has been ongoing in my group. I'm going to focus more on vaccination delivery by non-parenteral routes and what we've been working on. So next slide. So vaccination is an important public health strategy, as you've all known from this COVID epidemic being a prime example. So it's important due to basically reduction in global morbidity, mortality, and it's the most effective way to reduce disease and death from infectious diseases. So vaccines have eliminated some of the most dangerous and fatal diseases in human history. So far, we've used vaccines to wipe out or to like smallpox, and but also extremely close to eradicating polio. An additional 1.5 million deaths could be avoided, however, if global vaccination coverage improves. So next slide, please. So I mentioned I'll be focusing on pulmonary delivery, and respiratory diseases are amongst the leading causes of death worldwide. So the pie chart on the left here indicates a quarter of all respiratory deaths are associated with lower respiratory infections, of which pneumonia is the most significant. In addition, this also translates into hospital admissions. So almost half of respiratory admissions are attributed to acute infections. So again, including, you can see pneumonia being a large portion there as well. And episodes of infection are often a cause of additional exacerbations of lung diseases such as asthma and COPD. So this data is primarily in the EU, but it probably similar things across the world that you'll see. So next slide, please. So there's also economic burden associated with the disease, and it's been estimated to cost billions of dollars or euros in treatment costs and indirect costs with days lost to work, etc. On average, it's estimated to have an impact on costs to patients in the regions of about $20,000, which has most likely increased over the last 5 years. So again, the vaccine is good in terms of preventing, but one thing we don't look at or is an important consideration is also the economic burden that these diseases also cause. So next slide, please. So I mentioned pneumonia in the previous slides with regard to lower respiratory infections. Strep pneumonia is the leading cause of invasive and non-invasive pneumococcal diseases. So current vaccines do have limitations in coverage against the different serotypes. So a new vaccine could be inhaled to enhance mucosal immune responses and also have a greater coverage of serotypes would also be useful. I'll come to this shortly. So next slide, please. So if you look at the current licensed vaccine strategies, so these include, depending on where you are in the world, but you've got your PPV23, which was the initial pneumococcal vaccine, and this incorporated the polysaccharide from 23 of the common serotypes of strep pneumonia. That then, the later ones are conjugated vaccine, which contain the purified capsular polysaccharide antigen, and they contain from 7 to 10 to 13 common types of S. pneumonia, which are conjugated to a protein carrier. These are all administered by the intramuscular route, and as can be seen, they only protect against a limited number of serotypes, plus they also have limited mucosal protection as well. So next slide, please. So why is this an issue? Well, despite existence of licensed vaccines, strep pneumonia remains a leading cause of ill health and death, not only in children, but also the elderly and immunocompromised worldwide. And we've also seen cases now, if you read the literature of, with the current COVID situation of issues associated with these pneumococcal diseases. So current vaccines face several shortcomings, such as you got protection only against a limited number of serotypes. This can then also lead or induce serotype replacements. You get poor vaccine elicited in mucosal immunity, and there's also the high costs, so production, storage, transport, which limits affordability and availability, especially in low middle income countries. Hence, the development of a serotype independent pneumococcal vaccine could solve these shortcomings by conferring universal protection and reduced production costs. So in my laboratory, as my research group, we've been investigating protein antigens which are common to all serotypes of S. pneumonia. So next slide, please. So the rationale for aerosolized vaccination, i.e. delivery to the lung, is based on the following advantages over parenteral therapy. First, vaccination by inhalation avoids the need for disposal strategies for the large number of needles that are used in mass campaigns in developing countries. Secondly, it prevents the spread of bloodborne diseases such as hepatitis B and HIV, which can be transmitted by improper use or handling or disposal of the needles and sharps. Thirdly, administration of a vaccine via the aerosol route has less need for medical personnel or medical setting compared with administration by injection, and should facilitate vaccination implementation in developing countries. Another rationale for aerosolized vaccination is that it induces protection by exposure of the airways to antigens directly that affect the lung and cause diseases. So this can also be used for the likes of such as tuberculosis, pneumococcal diseases I've mentioned, as well as you can have others that we can use this route to induce mucosal protection, such as diphtheria, tuberculosis, measles, etc. The lung also has advantages which include antigen-presenting cells within the lungs, including the true antigen-presenting cells, your dendritic cells. So the lungs are also part of a mucosal-associated lymphoid tissue, which includes your bronchial, nasal, gut, and rectal associated lymphoid tissues. So this means the T cells can pass to other mucosal and systemic sites around the body, and then hence you not only get mucosal protection at the site, but can also get whole body protection as well at other mucosal sites. And systemic protection would also be initiated as well. Hence, airway mucosal vaccination may also present a potential approach for immunizing against agents that do not directly affect the lungs, okay, such as the human papillomavirus and hepatitis B. So this, as I mentioned, is because we can also get antibodies production in the serum. Administration can also occur via portable devices. So you've got your conventional dry powder inhalers or nebulizers, which are all easy to use. So next slide, please. In terms of nanoparticle delivery of vaccine antigens, it's worth looking at lung barriers and how these must be overcome to ensure effective vaccine delivery. So to deliver actives to the lungs, we want to target the respirable airways, and the size here needs to be between 1 to 5 micrometers. So here's the picture in the top left. However, for vaccines, I'm discussing nanoparticles, and these are generally less than 1 micrometer. And as dry powders, they have low mass and are exhaled as they don't travel far enough into the lung during inspiration. So how can we deliver these into the lungs? So I'll explain this in the next coming slides and what we have done in our lab. Also, it's worth looking at the lung physiological barriers. So picture on the bottom left, in the bronchi area, you can see this thick mucus layer, large epithelial cells, so it makes it difficult to penetrate. The deeper we enter the lungs, the anatomy changes. So you can see as we enter the terminal bronchi, the start of the respirable airways, the mucus layer is replaced with a thin layer lung lining fluid. The cells are smaller and makes it easier for nanoparticle penetration through here, and it can also access the dendritic cells, which are found in this respirable region. So hopefully this gives some rationale of why we're targeting the lungs, as well as the barriers and how we can overcome these. But the dendritic cells are true antigen presenting cells, and they are found throughout the lung. However, it had been demonstrated that the dendritic cells in the conducting airways are predominantly phagocytic, with some having antigen presenting cells, whereas those in the lower airways, so where I've just mentioned about the respirable part, are predominantly antigen presenting, hence the delivery required to the respiratory part here. Once the nanoparticles are deposited in the lower airways, they can diffuse and pass through the lung lining fluid, and they can be taken up by the dendritic cells. The DCs then change from immature to mature and process the antigen and present it on the surface via your MHC2 complex. From here, they can then travel to your lymph nodes via the afferent lymphatics, where they can interact with naive T cells. And this is the important part. So this is what makes the true antigen presenting cells, as they interact with naive T cells, which are new T cells not exposed to antigen before. Hence you get a new immune response. The T cells then migrate back to the lung, as well as travel to other mucosal sites. Next slide, please. So our approach is the design and development of a nanocarrier system incorporating the protein antigens for lung administration, and is to load them into microcarriers indicated here as the NCMPs. So the microcarrier is the 1 to 5 micrometer, so that will deposit in the respirable airways. It'll be made of a material that can dissolve and disperse within the lung lining fluid that releases the nanoparticles, and then that can diffuse through and be taken up by the dendritic cells. Next slide, please. So the first steps in development of the nanocarrier system involved optimization of the nanocarrier with the protein antigen. So here we went for adsorption approach to minimize the reduction interaction with the organic solvents, which can have a negative impact on the antigen in terms of like degradation, structural changes, etc. And hence that will have an impact on the uptake and process, and can have an effect downstream in terms of the immune response. The nanoparticles were produced using polymers developed in-house in LGMU. The method of nanoparticle production used the conventional emulsion method. So you have an antigen with surfactant PVA in aqueous phase, and it's mixed with the polymer in the organic phase, such as DCM. This forms your primary emulsion. This is then added to a second, larger aqueous phase under sonication, again with the surfactant, and this produces your double emulsion, and it's left over 2 hours for evaporation to occur. So there are many parameters to consider. This is shown in the right here. So you've got your parameters 1 to 7. All these parameters have an impact on the particle size, the particle charge, as well as the absorption of the protein onto the nanoparticles as well. So we optimized this using a model protein, and then those conditions were then applied to our particular protein antigen. So next slide, please. So now we've got our nanoparticle. So we then wanted to produce on nanocomposite microparticles, which incorporate the nanoparticles. And here we used a lab spray dryer, the Buki B290. And again, it underwent an optimization process in terms of investigating the ratio of nanoparticles to an amino acid. So we used an amino acid as the microcarrier, and within that was dispersed nanoparticles. This helps in the preservation and the activity of not only of the antigen, but also the nanoparticle, i.e. ensures it retains its size or as close to its size as possible. Moreover, we investigated the process parameters on the spray dryer, as indicated in the right. So the aim here was to achieve a maximum yield with low moisture content. So this is important for dry powders as it enables storage under normal conditions, avoiding any cold chain requirements and ensuring the nanoparticle and antigen stability and integrity is preserved. We also then investigated the aerosolization performance, so to determine how much and the quantity that's deposited within the respirable airways, and we investigated this in vitro using a cascade impactor. So next slide, please. So in terms of our optimization work, so figure 1 showed the mean signal to noise graph or particle size for each parameter. So here we used the Taguchi method to investigate the parameters. So the parameter with the largest range and corresponding rank indicates the relative importance compared to other parameters. So in this case, the letters A to E denote the experimental parameters, and numeric values denote the parameter levels. So here you got white diamonds indicate the maximum signal to noise ratio. So this was done with like 6 replicates, and using the Taguchi method, we had 18 experiments, and the optimum conditions inferred from a range, rank, and signal to noise response were determined. So here you can see you've got like A1 with the clear diamond on top. It'll be A2, B2, C3, D2, etc. Figure 2 shows the amount of the protein absorbed per milligram of nanoparticle for different concentrations. The absorption of the protein onto the nanoparticles is expected to be mainly driven by hydrophobic electrostatic interactions and hydrogen bonding. The absorption of the protein increased with increase in nanoparticle to protein ratio from 100 to 4 to 100 to 20, which was expected as you got more amount of protein there. Figure 2b suggests that the surface of the nanoparticle was saturated with the BSA, that's the model protein, after 1 hour, suggesting maximum absorption with 100 to 20 nanoparticle to protein loading concentration. Figure 3, we investigated a confocal microscopy was used to observe the interaction of the protein with the nanoparticle. So the microscopic images in the top shows the spray-dried nanocomposite microparticles containing the fluorescent nanoparticles. So red labeled, so using now red, and then the absorbed protein was a FITC green. The images show the FITC BSA was distributed where the nanoparticles were present, indicating their association. And then we did a SEM characterization to look at the shape, surface, morphology, and texture of the nanoparticles. So you can see they're slightly irregular and corrugated. And this also is useful in aiding in aerosolization. So again, this aids and ensures that you're maximizing the deposition into the lung. Hence, it will ensure that we have maximum availability of the nanoparticle and protein. Next slide, please. So from the preliminary studies using the model protein, we're able to determine the process parameters to produce nanoparticles and determine the optimum ratio and the mixing time for absorption. In addition, we optimize spray drying parameters to produce the nanocomposite microparticles of a suitable size for respirable deposition and hence delivery of these nanoparticles. So we now use these parameters to produce our vaccine formulation and determine their physical chemical characteristics and the efficacy of the formulation and the aerosolization properties. So next slide, please. So in table 1, you can see we achieved a size of 300 nanometers with approximately 19 micrograms per milligram of protein loading. So the size indicates the suitability of uptake by dendritic cells, being in the nanometer size. So a lot of work in the literature, and we've shown ourselves that sort of anything sort of 100 to 500 nanometers is sort of an optimum range. A reasonable yield in the NCMPs, the nanocomposite microparticles, were obtained, so over 50%. And you can see from the SEM pictures, so we got blank nanoparticles would be the shape to be irregular with corrugated surface, as explained. So the corrugated and regular surface ensures minimal adhesion contact between particles and hence the ease of dispersion for inhalation. Hence that can be seen here where we've got a high fine particle dose, so 60 micrograms is being delivered of the formulation, and the fine particle fraction is 74%. And you can see you've got your MMAD, so this is your mass median aerodynamic diameter. It's about 1.7 micrometers, so this also gives an indication of deep deposition into the lungs, targeting the bronchial alveolar regions. So again, we're maximizing our chance of delivering the nanoparticles to this region via the NCMPs and then delivering the vaccine. Next slide, please. So we then investigated the stability and structure and integrity. So we've got the protein, the PSPA, was released from the nanoparticle NCMP structure and is analyzed for primary and secondary structure using STS-PAs as well as your circular dichroism. So figure 1A indicates the PSPA released from the nanocomposite microparticles remained intact based on the presence of a single band between your 43 and 66 kilodalton. And no other bands of either small or large molecular weight were visible, indicating no degradation or aggregation. So this shows the protein remained intact during the absorption, the spray drying process, as well as the desorption, so release. The conformational changes of the PSPA released from the nanocomposites are also shown in figure 2. So the data for the spectra for the standard protein, so this is without doing any formulation, and the PSPA released. So indicated that the predominant structure of the protein was alpha helical, which compared to the standard, there was a slight decrease of about 13%. So this effect could be due to the absorption and desorption process during the process of absorption onto the nanoparticles, as well as maybe some release during the spray drying process. Next slide, please. We then looked at antibody recognition. So, we've absorbed onto the nanoparticles, we've undergone a spray drying. So has there been any additional structural changes that can impact on the recognition and then have a downstream effect on an immune response? So here the antibody recognition of the released PSPA from the nano composite microparticle was determined by the ELISA with a specific anti-PSPA monoclonal antibody and represented as a relative antibody recognition to the standard. So despite the decrease in the helical structure shown in the previous slide, this figure shows that the PSPA released from the NCMPs maintained its integrity with relative antibody recognition of almost equal to 1 compared to the PSPA standard. So this was encouraging, and it gives an indication that as a vaccine, hopefully we should get a good response. Next slide, please. So we then did the size, charge, shape, and size of delivery all affect dendritic cell uptake, and thus the immune response generated with nanoparticles, generally less than 500 nanometers, showing higher uptake and activation of dendritic cells, as well as inducing a stronger immune response. So here to show that and demonstrate our claims for effective dendritic cell uptake of approximately 300 nanometers, we used JAWS2 dendritic cells, which is a cell type, and we visualized the uptake by confocal microscopy. So the cell wall of the dendritic cells was stained with the Alexa flora, nucleus was stained with DAPI, and the nanoparticles contained the NAL red. So, and we observed under the respective green, blue, and red channels. So you can see from figure 1 on the top here, a typical split view of dendritic cells incubated with the nanoparticles for 1 hour in cell culture media and the PBS buffer respectively. So in the cell culture media, the NAL red fluorescence was detected when the polymer was co-incubated with the dendritic cells, indicating the presence of the nanoparticles within the dendritic cells. However, there are reports observed in the literature that the NAL red was released in the presence, or can be released in the presence of cell culture media and or transferred within cells. So in order to ensure that it was the actual nanoparticle that was incorporated, we then did the study with the PBS media. And you can see similar results there of the nanoparticle being uptaken. Next slide, please. So all of this so far shows good promise in terms of what we've done in vitro. So we've got good size, we've got good absorption, we've got good aerosolization properties, got good stability and integrity of the protein. So now we went on to do some animal studies. So mucosal immunization targeting the lungs was done with the resuspension of the nanocomposite microparticles, incorporating the nanoparticles with the PSPA. The purified protein was used as a control, and this was administered parenterally. So you can see here that's donated by the PSPA4 Pro SC. And the mucosal PSPA4 in the lungs as well. So mice received 2 doses of formulations with a 14-day interval, and anti-PSPA IgG titers were measured in the serum obtained from the blood samples collected after 14 days. So we observed a significant increase in the IgG titer in serum of mice immunized with the PSPA subcutaneously, and as well as within the nanocomposite microparticles, compared with saline after the first, so that's in figure A, and the second doses in figure B. You can see this negligible antibody response observed after mucosal immunization with the protein alone. So importantly, installation of purified PSPA induced a negligible immune response, indicating that the formulation of the protein in the nanoparticle is essential. Immunization with the nanocomposite microparticle PSPA4 elicited lower responses than the subcutaneous after the first dose, but a robust antibody response was seen after the second dose with higher titers. Both the subcutaneous and the nanocomposite microparticle PSPO elicited significantly higher anti-IgG compared to Ig2A, so indicating a bias towards a humoral response, a Th2 response. Next slide, please. So 21 days after the second dose, we also, the BALF was collected and we looked at the IgG and the IgA were measured. So here the nanocomposite microparticles with the PSPA4 showed significantly higher IgG in the BALF than in saline. So moreover, the subcutaneous induced a slight but not significant increase in IgG, so that's in 2A, whereas IgA levels were not increased in any of the groups, so that's in 2B. So this is expected to some extent, as strep pneumonia does secrete some enzymes that can neutralize the IgA, so it's not surprising to see results in where your IgA levels are quite low. Next slide, please. So we then went on to do efficacy of lung immunization was tested with a lethal challenge against a pneumococcal strain, which is listed there as the ATCC 6303. And this was delivered to lungs 21 days after the second dose. So partial protection was observed for mice immunized with subcutaneous. So that's your black square on the left-hand side in figure 1. About 50% out of the 6 animals that were used per group. But you can see with the nanocomposite microparticle, that's almost about 70%. So about 4 out of 6. So the bacterial load in BALF was also collected 12 hours after challenge and was analysed. So only animals immunised with the nanocomposite nanoparticle protein formulation had significantly lower pneumococcal load compared with the saline. Approximately 1 log reduction in the colony forming units was observed, so this is figure 2. You can also see quite a dramatic drop compared to subcutaneous as well. Although the overall survival after 10 days was similar in animals immunized with subcutaneous, as well as the nanocomposite microparticles, the group inoculated with the nanocomposite microparticles showed an earlier response in controlling the infection at 12 hours after challenge. So hence this is why we're seeing the significant drop in the colony forming units. So these results indicate, although immunization strategies induce similar final survival, lung immunization with protein-absorbed nanocomposite microparticles induce an earlier control of infection. Hence, it's possible that the higher IgG titers seen in the BALF after immunization with this nanocomposite microparticle formulation compared to the subcutaneous contribute to the earlier control of infection to the group. Next slide, please. So we also looked at the cytokines and chemokine content in BALF was also analysed. So you had levels of your different chemokines and cytokines were measured there as indicated. We observed differences between groups only for the pro-inflammatory cytokine IL-6 and the TNF-alpha, and for the neutrophil chemotraxins KC and CXCL1 and the MIP2 and CXCL2. Overall, Higher levels of these cytokines or chemokines were observed for the subcutaneous compared to the naive mice. These results are in accordance with protection elicited by immunization with the subcutaneous as well as the nanocomposite microparticle having different or progressing differently with higher inflammatory responses and the pneumococcal load in the PSPA group after 12 hours. Studies using naive mice have also shown elevated levels of your IL-6, IL-1 and the other chemokines in lung tissue starting at 24 hours post-challenge in animals that do not control pneumococcal infection. So next slide, please. So in summary, where are we up to at the moment? So we've successfully absorbed the pneumococcal protein onto our nanoparticles. We've retained the structural of the antibody, of the antigen after release. We've got good aerosolization performance. We've shown uptake into dendritic cells, and we've got good IgG response in serum and as well as in the bowel. We've also got lower bacterial load, and we've got pro-inflammatory cytokines, chemokines were detected in the bowel. Where we're up to now is we've now changed this formulation to include, actually I can do that on the next slide. So where we're up to now is we've managed to get some funding and to investigate this further. So we've developed a new method of making the nanoparticles which can be scaled up. We've now gone for an encapsulation rather than absorption, as additional in vivo work has shown encapsulation gave a better immune response compared to the better protection, compared to absorption. And we've also enhanced the aerosolization performance as well. So hopefully in the next conference I'll be able to present that data. We're just waiting for our publications to come out first. We've also got some industrial academic collaboration looking at some toxicology studies of these nanoparticle systems and the safety aspects. And we've also applied this technology to other lung delivery diseases and infections, such as delivery of antimicrobial peptides for treatments of lung infections. We've done gene delivery such as microRNA for treatment in lung cancers. Next slide, please. So finally, I would just like to acknowledge people who have been involved in this group. So there was the initial PhD students and recently the postdocs who have continued and modified this work further and progressed the work. We've got our collaborators in Brazil who are involved in the production of the protein antigen as well as the in vivo studies. Collaborations with the Trop School in terms of a clinical evaluation and using their input in taking this forward into possible human challenge models and clinical trials. And then my colleague Sarah in University of Central Lancashire for the circular dichroism. Next slide, please. So I'd just like to finish off with just putting a shout out. I am an editor-in-chief of Drug Development Industrial Pharmacy. And so this is a long established journal, which has an impact factor of 3.2. So we are looking for people to also join as associate editors. So if you're interested, please drop me an email. But feel free to have a look and see what type of topics and publications or the aims and scope of the journal, I should say. Okay, next slide, please. Okay, so that's the end of my talk, and sorry about the issues at the beginning, but thank you all for your for listening, and I'll be happy to answer any questions if we've got time.
Thank you, Dr. Imran, for your presentation. Does anyone having any queries?
Maybe I have a question.
Yes, you can ask
Uh,
Dr. Imran.
yeah, thank you very much for the very interesting presentation, Iman. Just one question about the nanoparticles. I don't know if you did any stability of the nanoparticles, because what I said that you use different techniques in order to manufacture nanoparticles, the spray drying, and you have another technique. There's a double emulsion. That's right.
Yeah, so in this work, the double emulsion was used to produce the nanoparticles, and then
Yeah.
we spray dry. So that's where the nanoparticles become incorporated with the amino acids to produce the nanocomposite microparticles. So then you have a dry powder. So that is then micrometer size. So we're able to aerosolize that and deposit in the respirable airways. The amino
Yeah.
acid disperses in the lung fluid, and then the nanoparticle would diffuse through the lung lining fluid and be taken up by the dendritic cells. In terms of stability studies, I haven't shown data here, but we have done some in where the final formulation, i.e. the spray-dried nanocomposite microparticle, so that contains the nanoparticle and the antigen. We have done some stability studies to look at, if we then redisperse the formulation, do the nanoparticles retain the same size? Is the antibody and antigen, is the antigen still retaining its stability and structure? So I haven't shown the data here, so that's the next part in terms of our future work and publications that will be coming out soon.
And the carrier you use in order to administer the nanoparticles, did you introduce the nanoparticles alone or mixed with the carrier, the inhalative administration?
Into the mice?
Yeah.
It was the full formulation, so it was the spray-dried nanocomposite microparticle.
Because will you in the future, is it you imagine to introduce nanoparticles alone by humans, or you have to mix them with the carrier such like lactose or?
No. So we don't mix with lactose. We use the amino acid.
Amino acid.
But yes, you can do nanoparticles alone, but then you have to ensure their stability. And the reason we produce a dry powder with spray drying is that it can then be stored as a dry powder in normal temperature and humidity conditions. So you don't have to store in a refrigerator or freezers or things like that. And likewise, depending on whether you want dry powder or nebulization, that spray dry powder can be given as a dry powder, or it can be dispersed in the nebulizer, and then the nanoparticles would be aerosolized.
Because I think that when you administer the nanoparticle alone, you will have some problems with the aggregation of nanoparticles. I don't know which size do you have. Is it less than 50 nanometers or?
No, these are about 300
nanometer.
Nanometers.
Yeah.
If you deliver them directly, the only way to deliver directly is with nebulization. And when you deliver via nebulization, your nanoparticles would be in the droplets. So your
okay,
saline solution that you use. So your droplet would be your sort of close to the micrometer size, and your nanoparticles would be in there.
Okay, thank you very much.
No problem. And any other questions?
Thank you, Dr. Imran, for answering the queries. Does anyone having any more queries?
I mean, my email's there as well, and I didn't say before, but we're open to collaborations, and also if you want to get in touch about the journal drug development industrial pharmacy, please feel free to drop me an email.
Sure, you will get an email from me. Thank you.
Good.
Okay, doctor. Once again, thank you for your presence and your presentation. Now let's move on to the next presentation. The next presenter is Dr. Ines Castagna from University of Calabria, Italy. Hello, Dr. Ines. Are you ready with your presentation?
Yes, I am.
Yeah, now I'm making you as a presenter. Kindly click on the screen button to share your screen.
Okay. Do you see that?
No, doctor. First, you have shared your screen, but now it is not visible yet. Now it is visible. Yeah, you can start with your presentation from presentation mode. You can move that GoToMeeting toggle.
It's okay now?
It's okay. You can start your presentation.
Okay. Thank you. So, thank you to everyone. Sorry, 1 minute. Alternative mouthwash formulation for oral care for delivering quercetin and mint oil in phospholipid vesicle. The oral cavity represents an important point of contact with the external environment and can act as a transit or colonization site for pathogens and external harmful substances. Today, the oral cavity is considered to host the second most complex ecosystem of the human body, after the gastrointestinal one. Thus, oral care provides an important protection, which in turn may improve the quality of life. As we know, quercetin is one of the most abundant flavonoids in nature, naturally present in food, including vegetable, fruits, tea, and wine. Quercetin is a pentahydroxyflavone, well known for its biologic and beneficial effects, such as antioxidant, anti-aging, anti-inflammatory, prebiotic, and metabolomic modulation activity. Essential oil, mainly located in agar plants, are a complex blend of lipophilic, liquid, and volatile compounds. Moreover, monoterpenes, sesquiterpenes, and phenolic compounds are present. Due to their chemical composition, show antioxidant, anti-inflammatory, and antimicrobial activity, mainly related to their ability to interfere with the functionality of whole proteins that are involved in the processes of transport of compound in the pathogen membrane. The use of natural antioxidants such as quercetin in formulation for external use represents an important strategy for defending skin and mucus from oxidative damages and for restoring their optimal condition. In addition, the association of natural antioxidants with essential oil can lead to interesting synergic properties, allowing a double protection effect both against oxidative stress and bacterial damages. Despite its promising beneficial properties, the low stability and solubility of quercetin limited its use in both pharmaceutical, cosmeceutical, and/or nutraceutical application. A model strategy to improve the stability and efficacy at skin level of drugs is their incorporation into nanocarriers. Is a versatile system capable of protecting the incorporated bioactives and increasing their effectiveness and safety. Among the different nanocarrier, phospholipid vesicles are the most promising, especially for skin and mucus delivery. They are mainly composed of phospholipids, amphipathic molecules with hydrophilic head and hydrophobic tail, which in water aggregates, forming a closed similar to that of cellular membranes. Thanks to their structure, lipophilic and hydrophobic bioactive can be intercalated in the lipid bilayer or in the aqueous compartment, given that in this study quercetin has been co-loaded in liposome enriched with twin 18. In this slide, I reported the composition of vesicles, which, as can be seen, are mainly composed of phosphatidylcholine, P19, and increasing amount of mean oil, and 15 milligram of milliliter of quercetin. Vesicles were prepared by direct sonication, a green approach in which the use of organic solvent is completely avoided. The main physiochemical properties, such as mean diameter, polydispersity index, and zeta potation, and technological properties like entrapment efficiency, storage stability, and stability against pH salvary were evaluated. Then, in addition, the evaluation of the biologic ability of vesicles, such as biocompatibility, protection against oxidative damages, and in vitro determination of antibacterial activity has been also evaluated. During the study, a pre-formulation test has been performed to evaluate the maximum amount of quercetin and mint oil co-loadable in liposome. The obtained vesicle loaded with 25, 50, and 20 and 100 milligram per milliliter of mint oil were the most suitable concentration capable of giving vesicle small in size, good dispersed, and highly negative charged. All formulation presents a slightly yellow color and transparent, containing small and homogeneous vesicles with pleasant organoleptic characteristic and associated to an intense mild aroma, therefore suitable to be used as a mouthwash for oral hygiene, for oral care. Cryo-temp analysis confirmed the presence of unilamellar, small in size and homogeneous system, and the storage stability study disclosed a high stability of the prepared vesicle dispersion. Indeed, all the formulation remains stable for all the monitoring time, one year. In the light of the possible use, also tested the conduct of the vesicle dispersion after dilution and incubation with stimulated saliva medium at pH 7. Then the dimensional parameters of the formulation were highly affected, showing an inversely proportional behavior with respect to the concentration of mint oil loaded in the formulation. Probably due to the hydrogen ion provided by the saliva medium that neutralized the negative group of phosphatidylcholine and breaking down the zeta potential. However, considering the low contact time between the mouthwash and the oral mucosa, this data do not seem to negatively influence the efficacy of the nanoformulation. Then, the most representative cell of the skin, keratinocytes, were used to evaluate the cytotoxicity of sample by using the MTT colorimetric assay. Results demonstrate that both quercetin and mint oil did not show a relevant toxic effect. Then, the ability of formulation to avoid damages caused by oxidative stress was evaluated, stressing keratinocytes with hydrogen peroxide for 4 hours, and in the meantime, treating them with dispersion or loaded liposome. We can see in the graph that oil in dispersion partially avoided the toxic effect of hydrogen peroxide, while its loading in vesicle ensured a better protection, leading the restoring of the healthy condition. Finally, The microbiological results suggested that the well-known antimicrobial activity of both payloads was improved by their incorporation in vesicle, but only when the higher amount of mint oil, 200, was used, as they were capable of inhibiting the growth of two representative pathogens of the oral cavity, the mutants and the acidophilus. Probably due this concentration of essential oil were not enough to stop the growth of pathogens. In conclusion, results confirmed the promising property of quercetin and mint oil liposome improved with twin AT, propylene glycol, and ethanol. They form a yellow transparent dispersion with a pleasuring aroma of mint, with a small vesicle, highly stable on storage. Their tolerant systems were highly biocompatible and capable of counteract oxidative and bacterial damages, and this formulation might represent an effective antibacterial antioxidant mouthwash for the conventional treatment of oral cavity disease. Thank you for your attention.
Thank you, Dr. Ince, for your presentation. Does anyone having any queries? I hope there are no queries. Once again, thank you, Doctor, for your presence and presentation. Now let's move on to the next presentation. The next presenter is Dr. Sofia K. Georgiou Stifris from Astytor University of Thessaloniki and University of Thessaloniki from Greece. Hello, Dr. Sofia. Are you ready with your presentation? Can you please unmute yourself? Hello, Dr. Sophia. I have made you as a presenter. Kindly click on the screen button to share your
Thank
screen.
you. Can you see? Okay. Let's begin. Hello. We want to thank the organizing committee for giving us the opportunity to present our work. This work has been conducted in the Laboratory of Pharmacology in Aristotle University of Thessaloniki and in the Laboratory of Veterinary Toxicology and Pharmacology in University of Thessaly, Greece. Okay. I always have this problem to change the slide. So,
Is Dr. Sophia?
no, I cannot change the slide. Okay. So, chimeric antigen receptor named CAR immunotherapy is based on the genetic engineering of patients' own lymphocytes to treat cancer. CAR is a fusion protein between the single-chain fragment variant named SCAV of a specific monoclonal antibody fused to intracellular sequencing for signaling of T cells. Indeed, the application of CAR-T immunotherapy in patients suffering with ALL and targeting CD19 yields more than 90% recovery rates. So, this is the structure of a CAR protein consisting of the extracellular part, that is the single-chain fragment variant, and the hinge, the transmembrane part, and the intracellular signaling sequences. There are several generations of CAR now. Here you can see the first 4 generations. In our work, we have applied the second generation, where 4-1BP or CD28 are connected to CD3Z. 4-1BP and CD28 are correlated with altered persistence of immune cells as well as bioenergetics. So, here are the first patients suffering from leukemias and treated with these innovative therapies. It is highly also important to mention that CAR-T cells are detected in the bodies of many patients years after the onset of therapy. So, Here we have the first such innovative products approved and entered market in 2017. Kymria is the first such product. It is estimated that in the near future we will have more such therapies available. So, of course, CAR-T immunotherapy is correlated with impressive anti-tumor responses. However, there are some safety-related issues to be resolved, like the cytokine release syndrome, named CRS, and the neurotoxicity. So fortunately, we have an alternative that is natural killer NK cells. NK cells are correlated with high tumor killing potential, low risk of graft versus host disease, and low CRS syndrome. Moreover, NK92 cells are a stable cell line of NK cells offered as an off-the-shelf product for application of CAR-NK immunotherapy. So these are the clinical trials that are ongoing, employing CAR-NK immunotherapy and targeting to treat both leukemias and lymphomas and solid tumors. What we have is the main way to equip the immune cells with the CAR sequences is the genetic engineering. Of course, it is correlated with some risks, like the insertional mutagenesis, the restriction in the length of construct, as well as the possible insertion of heterogeneous copy number. The other issue is the delivery platform to be used. We know about lentivirus, retrovirus, electroporation, and nanoparticles. Some of them needs to be improved. The alternative that is proposed is to use the in vitro transcribed named IVT mRNAs. So, IVT mRNAs is a prominent class of biopharmaceuticals aiming to deliver the genetic information based on the central dogma of biology. Almost all change in a night when the respective vaccines from Pfizer, BioNTech, and Moderna were approved. What about this technology? IVT mRNAs, upon their successful transduction into cells, they are translated into the desired protein based on the cell's own machinery. Looking beyond the pandemic, it is reasonable to say that IVT mRNAs is in the spotlight, becoming a key asset in modern medicine. So, IVT mRNAs have been introduced to us in 90s, but some issues were waiting to be resolved, like the immunogenicity and the instability. Of course, the selection of the proper delivery system remained an issue. Nowadays, we know that there are several chemical and structural modifications to be performed in order to increase the translational efficiency of IVT mRNAs, as well as to decrease immunogenicity, as has been proposed by Carico and Weissman. The modifications can be shown here in the slide, and many of them have been already introduced into our constructs. So, this is the technology behind and the idea. We have the linearized vector in order to produce the IVT mRNAs with all these modifications, and the packaging system in order them to be introduced into the cytoplasm of cells and to be translated. The applications, as you can see here, are numerous, and of course all we know about the mRNA vaccines against SARS-CoV-2. To choose another delivery systems is a challenge. So, protein transduction domains named PTDs or penetrating peptides, CPPs, are small in length peptides able to traverse almost all membranes and at the same time to transport many cargoes like proteins, plasmids, and so on. as already we have mentioned, the selection of the optimum delivery system is the challenge. Here we propose the exploitation of PTD technology. PTD can be used either not covalently or conjugated covalently as initially proposed by our research group. So, we have designed a novel IVT mRNA delivery platform via which the selected PTD is covalently conjugated to any desired IVT mRNA. The methodology behind has already been published in a Greek patent in May 2021 and the international patent pending. So, this is the aim of our study. We want to construct two different IVT mRNAs. Both harbor TAF1E, that is the single-chain fragment variant for RBB, and each one will contain either CD28 or 41BP. Both IVT mRNAs will be covalently conjugated to PTD in order to be stabilized and transduced into NK92 cells. These car-engineered NK92 cells will be our effector cells in order to be co-incubated with our target cells, HSC3 cells. HSC3 is a cell line of oral cancer. Oral cancer has high mortality rates and poor prognosis, as well as is associated with elevated levels of RBB expression. So, we are moving forward to the experimental procedure. It is separated in 7 steps. Each one will be analyzed as well as with the results acquired. The first 3 steps was to clone the desired constructs into appropriate vector for the in vitro transcription, the production of IVT mRNAs, and then the covalent conjugation to the selected PTD. These are the 2 constructs we have. Both of them contain the 5 UTR, TAF1E, CD3Z, high epitope, and 3 UTR. Each one will contain CD28 or 41BP, named for our convenience CAR-A and CAR-B constructs. So the first step was to add a overhang in our constructs in order to be cloned in PGEM TAF vector selected because it possesses the TAF7 promoter. Here we are the successful cloning, and the clones were picked up. Then, via diagnostic digestions, the proper clone with sense orientation was selected. This was for CAR-A construct, in analogous way for CAR-B construct, as we can see. And again, the clones with the proper sense orientation were picked up. So both constructs were verified by sequencing for their sequence integrity and proper orientation. Then the linearization took place in order to be ready for the in vitro transcription. The in vitro transcription took place in presence of anti-reverse kappa analogues, and after its termination, poly-A polymerase was added in order to add more than 100 adenines, as you can see in the gel electrophoresis here. So, then the covalent conjugation of PTD took place. Our confirmation was the band shift assay as shown in the gel here for both constructs. We are moving forward to the 2 next steps. We have noticed that PTT-IVT mRNAs were significantly protected to the action of RNAs for both constructs compared to the NECT IVT mRNAs. This was important. So we are moving to the intracellular transduction experiments. A complement to K92 cells and the PTD-IVT mRNAs, we used K562 cells, that is a cell line easy to be transfected, as well as lipofectamine, that is a well-known reagent for transfection. So in our first experiment, we used K562 cells transfected in presence of lipofectamine with either construct. By RT-PCR, we could detect intracellularly both constructs via using two different sets of primers, amplifying both a short part of our IVT mRNAs and the whole IVT mRNA. So, in the next experiment, again in K562 cells, now PTD-IVT mRNAs, both of them were detected in RT-PCR with both set of primers. We are moving forward to K92 cells, that is the desired cell line. It's an IL-2 dependent cell line. Okay, and our first goal was to see whether whether or not PTD-IVT mRNAs impair cell growth. As you can see, PTD-IVT mRNAs were found not to impair significantly cell growth, as shown by reverse microscope. K92 cells tends to grow and form spheroid-like structures, as well as by assessing cell growth. Moreover, we did not detect cell death by using a fluorescent probe and flow cytometry compared to the gray bar, that is K92 cells treated with oxorubicin, a cytotoxic agent. So we were ready to perform our quantitative analysis in the copies of PTD-IVT mRNAs transduced intracellularly. For this purpose, we construct a standard curve via using serial dilutions of cDNA. In this first experiment, we can see that PTD-IVT mRNA CAR-A is transduced intracellularly in high levels compared to our negative controls. Between them was the NECT IVT mRNA. Next, we have the time course analysis in K92 cells, showing that PTD-IVT mRNA was transduced into cells since 2 hours of exposure, then remained intracellularly via biphasic mode, and then detected for up to 120 hours post-transduction. In the next set, we have the expression analysis into the respective proteins. Again, in K562 cells treated with lipofectamine and either construct and Western blot analysis against CD3Z, we can see that both constructs were expressed into the desired proteins at 35 kilodalton. Subcellular fractionation of cells complemented by Western blot analysis showed to us that CAR-B protein is located mainly in the membrane fraction, as expected. In this experiment, actin was used as a loading control, but also as a marker for protein purity. So we are moving to K92 cells in both ways, and more importantly, via PTD-IVT mRNA technology. CAR-A protein was expressed again at the desired molecular weight. In the time course analysis, in K92 cells transfected with PTD-IVT mRNA for different time intervals, CAR-A protein was expressed quickly and then remained intracellularly for up to 72 hours. So we are moving in the next experimental procedure, that is to co-incubate the PTD car-engineered Nikkei 92 cells as the effector cells with HCC3 as the target cells. The first thing that we want to check is the well-known capability of Nikkei 92 cells to be cytotoxic to K562 cells. For this reason, after the co-incubation, we performed the trypan blue exclusion assay in the pool of cell populations. As you can see in the gray bar, the cytotoxic potential of Nikkei 92 cells was great. Then we are moving to these core experiments. These experiments are ongoing, but this is the general sim. HCC3 cells need to be seeded. Nikkei 92 cells are transfected with PTD IVT mRNAs for different time intervals in order to be then washed and added at the bottom of HCC3 cells in different ratios of effector to target cells, ranging from 10 to 1 to 20 to 1. In this time, the co-incubation period begins for different time intervals again. Then, the trypan blue exclusion assay is employed in different fractions, both in the cells in the supernatant and in the cells remained attached. Of course, for consistency, we have all our controls, that is HCC3 cells culture alone, as well as Nikkei 92 cells untransfected, and those transfected with our constructs. All these controls were also assessed. Here we can see the result of an experiment, including the transfection of cells, of Nikkei 92 cells, as well as the co-incubation of the cell population for 24 hours. The ratio was at 10 to 1. As you can see, Nikkei 92 cells mock untransfected are able to provoke a percent of cell death in HCC3 cells. Our car, the gray bar, our car engineered Nikkei 92 cells increase this cell death by almost two-folds. HCC3 cells culture alone characterized by minimum cell death. This is the tendency for attached cells, and analogous tendency was recorded for the suspension cells. So, in the next trial of these experiments, the transfection time was reduced to 7 hours. The ratio was increased to 20 to 1, and the co-incubation period was shorter to 4 hours. Again, we recorded the same tendency. Next, when all these parameters was the same, but the co-incubation period was extended to 48 hours, we could record the same result. So, to sum up, from all these results, PTD technology was successfully exploited for the rapid, efficient transduction and translation of the stable IVT mRNA encoding the CAR molecule into Nikkei 92 cells. CAR Nikkei 92 cells have shown efficient cytotoxicity over HSC3 cancer cells, leading to encouraging results for the application of our novel PTD-IVT mRNA delivery platform for CAR immunotherapy. In the near future, we plan to investigate alternative methods for the evaluation of CAR Nikkei 92 cytotoxicity. Also, we plan to test our CAR-engineered Nikkei 92 cells in another cancer cells as target, like the breast cancer cell lines. So, this research was funded by European Union. I want to thank you for your attention, and please ask me whatever you want.
Thank you, Dr. Sophia, for your presentation. Does anyone having any queries? I hope there are no queries. Once again, thank you, Dr. Sophia, for your presence and presentation. Now let's move on to the next
Thank you.
presentation. The next presenter is Dr. Judith Ovidi from Research Center of National Sciences, Hungary. Hello, Dr. Judith, are you ready with your presentation?
Yes, I am.
Okay, now I'm making you as a presenter. Kindly click on the screen button to share your screen.
Okay.
Your screen is visible. You can start your presentation from the first slide. And can you please unmute yourself? Hello, Dr. Judith, you are in your... Hello, Dr. Judith, we are not able to hear your voice. Can
Yes.
you please unmute? Yeah, now you can
Now, do
start
you
your
hear me?
presentation. Yeah, okay, you can start your presentation now.
Oh, okay, thank you very much. First of all, I would like to thank the organizers for the opportunity to present our results concerning the role of tubulin polymerization promoting protein in synucleinopathies and its possible therapeutic potential. With the aging of societies, neurodegenerative disorders became more and more common, causing serious social and economic problems. The development of such conformational diseases is a multi-step process where unfolded, misfolded proteins enter into aberrant protein-protein interactions, resulting in protein aggregation. The aggregates forms inclusion bodies, leading to the selective loss of cells in various brain areas. In the case of Parkinson's disease, the disease is characterized by intracellular Lewy bodies and Lewy neurites in neurons, while in the case of multiple system atrophy, glial cytoplasmic inclusions can be found in the oligodendrocytes. Parkinson's disease patients suffer from classical motor symptoms as well as non-motor features. Multiple system atrophy patients also display Parkinsonian features, but it is a more rapidly progressing disease. The motor impairments of Parkinson's disease can be attributed to the loss of dopaminergic neurons in the substantia nigra pars compacta. Currently, there are only symptomatic treatments. The gold standard drug is the L-DOPA or levodopa, which relieves the symptoms by the replacement of the lost dopamine. The major goal of Parkinson research is to develop disease-modifying therapies which can slow down or stop the underlying neurodegenerative processes. The Parkinson research focuses mainly on alpha-synuclein, the hallmark protein of synucleinopathies. Alpha-synuclein is a disordered protein which is expressed predominantly in neurons. Structurally, it comprises three regions. The N-terminal region is involved in lipid binding. The highly hydrophobic central region is responsible for the aggregation of the protein. The unfolded acidic C-terminus displays chaperone activity, and it seems to play a role in preventing synuclein aggregation. The disordered synuclein is able to adopt structurally unrelated conformations. its physiological function is still not well understood. It has been proposed that alpha-synuclein is involved in neuronal plasticity modulation, synaptic vesicle pool maintenance, and dopamine metabolism regulation. Previously, the alpha-synuclein aggregates were considered the most toxic species, but accumulating evidence indicates that the small, soluble, homo and hetero oligomers of synuclein are the real culprits for synuclein toxicity. We have identified a brain-specific protein, tubulin polymerization promoting protein, P25, which induces synuclein aggregation, and it also a hallmark protein of synucleinopathies. Similarly as alpha-synuclein, P25 is also a disordered protein. The unfolded N and C termini are straggling a highly flexible middle core segment. This core region involves different binding motifs, for example, a zinc finger and the GTP binding motif. P25 induces the formation of synuclein oligomers and protofilaments, and it is co-enriched and colocalized with alpha-synuclein in the inclusion bodies. In the case of Parkinson's disease, in Lewy bodies in neurons, while in multiple system atrophy in the glial cytoplasmic inclusions in oligodendrocytes. Tubulin polymerization promoting protein was designated on the basis on its in vitro and in vivo functions in the laboratory of Professor Judith Ovadi, and now the protein can be found under this name in the different gene and protein databases. In vitro, it induces the polymerization of tubulin into microtubules, normal, aberrant, bundled microtubules, and tubulin aggregates. In vivo, it is aligned along the microtubule network at low expression level. However, at high protein expression, it is able to form perinuclear cage or aggresomes. In HeLa cells, the acetylation level of the microtubule network is relatively low. Surprisingly, we found increased microtubule acetylation in P25 transfected cells. The binding of the protein to the tubulin deacetylase enzymes HDAC6 and sirtuin-2 resulted in increased tubulin acetylation, which, along with its bundling activity, influence the stability and dynamics of the microtubule system as well as the microtubule-derived cell motility. The dynamic microtubule network plays crucial roles in cell division, intracellular trafficking, as well as, at pathological conditions, in aggressome formation. Its early destabilization and altered microtubule dynamics has been observed in Parkinson's disease. Therefore, the microtubule network is also a possible drug target. In normal brain, P25 is expressed in oligodendrocytes. The expression of this protein is enhanced in the projections during the differentiation. It colocalizes with the microtubule network. P25 expression is crucial for the differentiation of oligodendrocytes because it stabilizes the microtubule network. The differentiated oligodendrocytes are the main constituents of the myelin sheath around the axons, which is indispensable for the proper functioning of the central nervous system. P25 participates in different pathological processes leading to distinct central nervous system diseases. Multiple sclerosis is a chronic inflammatory disease of unknown origin, which causes lesions in the ensheathment of axons. We found loss of P25 positive oligodendrocytes in the brain, as well as increased P25 level in the cerebrospinal fluid of multiple sclerosis patients. In oligodendroglioma, which is a malignant tumor type of oligodendrocytes, significantly decreased P25 expression was observed. As I mentioned earlier, P25 colocalizes with alpha-synuclein, and these two proteins are co-enriched in the inclusion bodies in synucleinopathies due to the cell-to-cell transmission of the proteins. Synuclein and P25 are challenging drug targets. In the case of globular proteins, the rational drug design is based on the well-defined 3D structure of the proteins, and well-established methods are available. However, the intrinsically disordered proteins display high conformational plasticity and participate in several interactions. Therefore, in spite of combination of computational and experimental methods, their successful targeting is still limited. Both synuclein and P25 are disordered proteins. Moreover, they are moonlighting proteins, displaying physiological and pathological functions as well. Therefore, none of them is an ideal drug target. But we can exploit the fact that the two proteins are expressed in distinct cell types in normal brain, synuclein in neurons, while P25 in oligodendrocytes. Their co-assembly occurs only at pathological conditions. Therefore, we have suggested a new innovative strategy, namely targeting the interface of the pathological synuclein P25 complex without affecting the physiological interactions of these proteins. In order to achieve this, first we had to identify the segments involved in complex formation. First, we compared the function of the full-length P25 with that of the double-truncated form. This is a termini-free form which corresponds to the highly flexible middle region. Theoflavin T fluorescence assay was established to follow synuclein aggregation. We found that both the full-length and the double-truncated P25 displayed similar alpha-synuclein aggregation promoting potency. In Chinese hamster ovary cells, the double-truncated P25 displayed an even higher alpha-synuclein aggregation promoting potency, probably due to the fact that the double-truncated form is unable to bind to the microtubule network. These and other data indicates that the C-terminus of the protein is involved in its interaction with tubulin, while the middle core segment of P25 is responsible for synuclein binding. We carried out NMR studies as well. Alpha-synuclein did not cause large structural changes in the labeled double-truncated P25. However, we found significant shift of some amide residues indicating their direct involvement in the binding. These data corroborated that indeed the middle segment of the protein is responsible for synuclein binding, and we identified these two segments. Are these segments responsible for the formation of the pathological complex? To investigate this, we carried out further binding studies using several deletion and truncated P25 mutants and peptides. We found that these P25 species displayed similar binding potency to alpha-synuclein, except one, This is the double truncated double loop form. So, rather surprisingly, we found that the binding segments can be replaced by other ones, which ensures exceptional functional resilience. Our next question was whether this characteristic also refers to the pathological, to the partner protein, alpha-synuclein. Therefore, we used different synuclein mutants and peptides to identify the synuclein segment involved in P25 binding. We found that the C-terminus of synuclein displayed similar binding to P25 in contrast to synuclein 1 and 120. Although synuclein is also a disordered protein displaying high conformational flexibility, we found that the deletion of the last 20 residues completely abolished its binding to P25, indicating that this segment is involved in P25 binding in the complex formation. This scheme summarizes the identified binding segments involved in the pathological P25 alpha-synuclein complex. Next, we applied bimolecular fluorescence complementation technology using Venus vectors to investigate the dynamic heteroassociation of the full-length P25 and alpha-synuclein as the pathologically relevant forms in HeLa cells. The BIFT technology exploits the intracellular interaction of the labeled proteins, which results in the close proximity of the two segments of the split Venus protein. Thus, it produces a fluorescence emission, a green BIFT signal. Previously, as I mentioned that in the transiently transfected HeLa cells, the full-length P25 align the microtubule networks, while the double-truncated form distributed homogeneously within the cytosol. Using BIFT's technology, we provided evidence for the heteroassociation of P25 and synuclein at cellular level. To investigate the dynamic nature of the heteroassociation and the availability of this method for drug testing, we also added unlabeled P25 to the cell, which indeed decreased the BIFT signal as shown by the quantification of the fluorescence emission. The data at cellular level underlined the results obtained in vitro using human recombinant protein and also showed the suitability of this method for testing possible anti-Parkinson drugs. What are the consequences of the inhibition-destruction of the pathological complex? What will be happen to synuclein and P25 liberated from this complex? The two major protein degradation pathways responsible for maintaining the protein homeostasis in cells are the ubiquitin proteasome system and autophagy. The ubiquitin proteasome system degrades mostly proteins, typically short-lived and disordered ones. Autophagy is responsible for the degradation of long-lived proteins, aggregated ones, as well as cellular organelles. It has been shown that both synuclein and P25 can be degraded by autophagy and the ubiquitin proteasome system. Therefore, we investigated the effect of the heteroassociation of synuclein and P25 on their proteolytic degradation. In this experimental setup, we added the proteins to the medium of the cells, and after their uptake, we determine the protein levels by Western blot technology. Interestingly, we found that the synuclein level is increased in the presence of P25 as compared to the synuclein alone. In the case of P25, there was no difference whether the protein was added to the cell in the presence of synuclein or alone. We also applied different inhibitors and activators. Rapamycin is a potent inducer of autophagy. The rapamycin-induced proteolytic degradation of synuclein was less effective when the synuclein was in the presence of P25 as compared to the synuclein alone. These results indicate that the heteroassociation of synuclein with P25 inhibits both their autophagy and the ubiquitin proteasome system-derived elimination. DJ1 is a multifunctional protein with antioxidant properties. It is able to protect dopaminergic neurons against Parkinson's disease. Therefore, we investigated the effect of DJ1 on the P25 synuclein complex. In this experimental setup, we also applied BIFT technology, but we co-transfected the cells with the BIFT plasmids as well as with mCherry DJ1, which allowed us to visualize the effect of DJ1. We found a decrease in the intensity of the BIFT signal. We also found an increase in the proteolytic degradation of synuclein complex with P25 in the presence of DJ1. These results indicate that DJ1 is able to abolish the inhibitory effect of P25 on synuclein degradation. To summarize our results, I would like to mention that synuclein and P25 are challenging drug targets. However, the dissimilarity of the binding motifs involved in the physiological and pathological interactions of P25 has significant innovative impact. Therefore, we identified and validated the interface of the pathological P25 synuclein complex as a specific drug target. And we suggested this innovative strategy for the development of anti-Parkinson drugs. This can be carried out preferentially by drug-like agents such as fordomaz, based on, for example, on the identified alpha-synuclein fragment. Then the excess P25 and synuclein can be degraded by the autophagy or the ubiquitin proteasome system. Finally, I would like to thank my co-workers and our collaborators for their contribution, and thank you for your attention.
Thank you, Doctor, for your presentation. Does anyone having any queries? I hope there are no queries. Once again, thank you, Doctor, for your presence and presentation. Now let's move on to the next presentation from Dr. Uterli from CIO University Hospital of Bonn, Germany. Hello, Dr. Uterli, are you ready with your presentation?
Hello, can you hear me?
Yeah, you're audible. Now I'm making as a presenter. Kindly click on the screen button to share your screen. Hello, Dr.
Yeah, I sorry, I can't share my screen.
Hello, Dr. Italy. Maybe our you have a network issue. We are unable to hear your voice clearly. And also we are unable to see your screen.
Mhm.
Hello, Dr. Italy. May I know the issue you are facing? Hello, doctor. Are you there?
Can I email to Peter or
Hello. Can you please click on the screen button in your GoToMeetings app? Hello, Dr. Italy. May I know the issue you are facing?
can I send email to Peter on? Okay.
Click on the screen button to share your screen, doctor. Your screen is shared, but you have not displayed anything. I hope so.
Oh, sorry, I can't share my screen right now. I go to Peter. Please, my,
Hello, Dr. Italy. We are unable to hear your voice clearly. I think there's a network issue from your end. Can you please leave the session and join again? Hello, Dr. Italy, your screen is shared. Kindly open your presentation and start your presentation. We're able to see your screen. Please open your PPT and start your presentation. Hello, Dr. Italy, can you please check your internet connectivity? Yeah, your PPT is open now. Hello, Dr. Italy, are you there? Start your presentation, Dr. Italy.
Yeah.
Your voice is not clear. Can you please check your mic connectivity or network connectivity and start your presentation? Your screen is visible.
I
It is
sent
perfect.
the email.
Your screen is visible. Your presentation is perfect. Can you please check your network connectivity or mic? Your voice is not clear.
I can't
Okay.
the PD now. Okay. Can you
Start your presentation, Dr. Italy. Your screen is visible.
still see my screen now?
Yes, your screen is visible. You can start your presentation.
Oh, okay. Um, and thank you very much. And without the PC data, um, when university started, the topic might not clear. Clear. Then can
I'm sorry, but I think we have to move to next presentation because we cannot
no, they are
clearly
really.
hear.
I don't know how to regulate. Um,
Hello, Dr. Italy. Can you please check your mic or your network
can you,
connectivity? We
can you
are
hear
unable to hear
me
your voice
now
clear.
or something wrong with my microphone?
Your voice is not clear, Dr. Italy. Can you please check your network connectivity or your mic settings?
It's okay.
Your voice is not clear.
Okay. for my friend.
If it is possible,
my
can you come from another device to
voice.
join the? No, your voice is not clear. There's a lot of
My
disturbance from your end.
talk today is combination of cytokine induced.
Hello, Dr. Italy. Can you please change your device or check your internet connectivity? Meanwhile, we'll move on to the next presentation. The next presenter is. Okay, meanwhile, we'll move on to the next presentation. The next presenter is Dr. Yavuz Salim Hishri from Istanbul Consulting Group Managing Partner from Turkey. Hello, Dr. Yavuz. Are you ready with your presentation?
Yes, I'm ready, happy to present. If you can give me opportunity to share my screen,
As
I'll
Doctor, I have
be
made you
honored
as
to
a present.
present.
As Doctor, I have made you as a presenter. Kindly click on the screen button to share your screen.
Sure, sharing right now. Thank you so much. I want to thank everyone for this beautiful meeting. Look forward to having a great conversation. I will make it full screen. This is Dr. Yavuz Selim Sulay. I'm a physician, entrepreneur, and venture architect. I'm the chairman of Istanbul Consulting Group
Sorry for the
and honored
interruption,
to
Dr.
present.
Yavuz. Okay, okay, sorry. You can
Yes,
continue your
honored to
presentation.
present. No problem. Honored to present to this distinguished group. And today I will talk about the strategic trends on biologic and biosimilars in the United States and Europe. As you all know, it has become a very important topic. I just wanted to highlight a little bit about myself. I own 2 television channels. I have a management consulting group and also own 2 finance groups. One is in crowdfunding, another one is in Angel Investors Network. And also I work with Rutgers University in Innovation and Education Center in New Jersey. And also I'm the president of Society of Physician Entrepreneur in Turkey. And honored to be here with this distinguished group. I especially want to highlight that you're all welcome to Turkey anytime, and we'll be honored to host all of you. These are my clauses in terms of conflict of interest. I am, currently getting money from these organizations, and I openly disclose all my conflict of interest. I worked in the United States, worked with Ipsen, French company, Teva Pharmaceutical, Forest, KV, Baylor College of Medicine, Chess Foundation, and other groups. So I would like to disclose all my, financial ties before getting to my presentation. Today I will talk about the biologics and biosimilars, and want to give you a highlight on biosimilars, and then talk from the originator firms for front line of the biosimilar competition and the key biologic brands facing competition and the extent of their exposure for key companies and how they are actually defending it, and the current landscape starting from 2017 to 2025, the biosimilars in the US and Europe. That's what I want to highlight today. So in the United States, there are 3 definitions for biologic medicines: the reference medicine, the biosimilar product, and the interchangeable product. We all know generics anyway, but I think the most important point is the most regulatory bodies only have 2 definitions compared to the United States. It's either biologics or biosimilars. So this is something I want to highlight, which is different in the United States. And this is very important point I want to highlight here. When we look, we all know the Pareto rule, which is the 80-20 rule, but actually 2% of the people are using biologic products now in the United States, but 40% of the spend for these 2% goes to biologics. So in terms of the amount that is the cost it's taking, you can think and understand how it impacts the budget for biologics. And we have aspirin, which has about 180 daltons. We have insulin, about 5,800 daltons. We have erythropoietin with 30,000 daltons. And Now the monoclonal antibodies are around 150,000 Daltons. So these are the relative molecular masses of these products. And when we look at the biosimilars, with the real evidence, with the right safety, efficacy, and quality combination, you can see that biosimilars are also a good option for patients. And over the last 10 years, more than 700 million days of patient experience have been collected with biosimilars. And to date, there is no new safety or efficacy concern. I want to highlight the 5 key characteristics that really characterize the biosimilars, how they can enter the market in a rapid way or in a delayed way. And when we look at it in a delayed way or rapid way, there are different strategies. One of them is indication. If you have an indication and you have most or all indications on label, then you can enter the market much faster than if you have a key missing labeled indication. Chronic versus episodic treatments. If you are an episodic treatment, a short-term treatment, you can enter the market faster as a biosimilar compared to a chronic treatment. Of course, the payment of these drugs are important. And when we look at the payer preference, they prefer biosimilars, and they also look at the parity with other biosimilars. And these products enter the market faster than the products which have a brand preferred and a preferred biosimilar strategy. Also, I want to highlight the objective versus subjective measure. If you have an objective measure, you are able to enter the market much faster than a subjective measure. And finally, the interchangeability is a reason in the market adoption of biosimilars. If there's a low need in terms of physician or hospital requirement, you can enter the market faster compared to a retail or referral infusion, which is more complicated. Also, I want to highlight some of the roadblocks of the current biosimilar. We have a theory, and we also have the actual utilization, and there are some roadblocks when we look at them. Some of them are in the United States, the separate J codes which are resolved, the Part D coverage of gap discount in the United States is resolved, and the pass-through status also remains for now. I think the biggest hurdles for biosimilars in US market is the reimbursement model, the payer preference, like the rebate trap, the provider and patient education, interchangeability, and discount pricing. And these are some of the hurdles we see as products enter the market. But I want you to all remember that when the Hatch-Waxman Act in 1984 has passed, which is the basis of generic drug market today, at that time, 35% of the top-selling brand drugs had expired patents and generic competition. So there was only 13% generic adoption. Now, fast forward 2022, almost 9 out of 10 of all prescriptions in the United States are filled with generic drugs. So there is almost 97% generic adoption. So even though there is a slow adoption in the US compared to Europe, we should not forget that for the generics, this has happened, even though it took around 30 years to reach almost majority of the market. And, you know, we need to learn from the generics in terms of the limiting factors of generic adoption, the brand loyalty, the brand market size, the drug characteristic, the regulation that promotes the generic substitution and reducing the market barriers, and the brand company strategy to minimize the impact of generic adoption are very important. Now, I want to highlight some of the big pharmaceutical companies, the key originator companies with blockbuster biologicals whose market exclusivity has either expired or will soon expire, like AbbVie, Amgen, Sanofi, Roche, Johnson & Johnson, and Lilly. Let's look at the 2000 sales of some of the biosimilar competition for key products. The Humira was in 2017 around 18 billion, Mabtera, Rituxan around 7.5, Remicade, Herceptin, Avastin, Enbrel, Lantus, Neulasta, Humalog, Epogen, Arenax, and Neupogen. So these are the top products that are facing biosimilar competition with large sales amount. And some of the extent of exposure these key firms having are Amgen, Johnson & Johnson, AbbVie, and Roche. And let's look at Amgen. When we look at Amgen, the current sales versus the projected sales in 2025, we see almost about 50% loss from 5.4 to 2.7 for Enbrel, from Neulasta 4.5 to 2.7, for darbepoetin alfa from 2 to 1.2, for Ipogen from 1 to 341, about 70% loss, for Neupogen about 40% loss from 550 to 344. So you see that there is a significant loss in the extent of exposure for Amgen. Also, when you look at Enbrel, Neulasta, Neupogen, Epogen are all facing biosimilar competition. That's why Amgen is hedging by building its own portfolio of biosimilars, which is expected to be a key growth driver for the company in the coming years. And this is just the Amgen analysis of how the evolution may take place by 2025 and how they're actually balancing their loss in biosimilars. Similarly, the Johnson & Johnson analysis is also very interesting. When you look at the 2000 sales of Johnson & Johnson with Remicade, you will see about 40% loss, 42% loss from $6.3 to $3.6 billion. And the Procrit, the epoetin alpha, also has about 68% loss from 970 to 330 million. And Stelara from 4 to about 4.6. There you can see actually an increase with the launch as well. So this is very important because when you look at AbbVie also has with Humira, which is the #1 product, projected sales of from 18 to 14, and a total pharma sales increase from 28 to 47. So companies with multiple biosimilars that are launched in European market, they find ways to actually compensate with other innovative products. Roche has also a very important trajectory for Avastin. From 6.7 to 2.7, you will see projected sales and change, which is about 60% loss. For Herceptin, from 7.1 to 3.7. For Rituxan, from 7.5 to 2.8. So these are important trends we see. So how are these large companies actually defending their top and bottom lines? #1 is the patents. They use different patent strategies to protect with different patent litigation, delayed biosimilar entrants, and they also utilize some life cycle management projects. And at the end, if they see they cannot beat, then they join actually. So as you can see, for the Boehringer Ingelheim, Biogen, Pfizer, Merck, and Amgen, model, they actually either join or also go head-to-head competition, and then if they go lower, they also go lower in terms of pricing. At the end, the defense tactics is also weaponizing patients. There are campaigns in US and Europe also, there's less susceptibility, but they are utilizing patients with the doctors and claiming, for example, you and your doctor worked hard to find Remicade. So these biosimilar medicines and switching them, they are giving some information and utilizing the patient power in advertising in campaign. At the end, another way is optimizing the R&D pipeline and replace the expiring products, next generation product development, new technologies and partnerships. And as I mentioned, for the defense, they grow a strong patent thicket and tying customers with discounts, bundles, rebates. They utilize lifecycle management, make or sell biosimilars, compete on price, and target patients, and most importantly, which is also the core of this meeting, innovate, innovate, innovate. And when we look at United States versus Europe, they are around, Europe is much more rich in terms of biosimilars compared to United States. Around 60 biosimilars launched for drugs, including Sanofi's Lantus, Amgen's Epogen, Pfizer's Enbrel, again, Neupogen, Neulasta, Remicade, Rituxan, Herceptin, and Humira, versus in the US, Filgrastim, Infliximab, and Pegfilgrastim, and Nivastim. So when we look at US, there is a more limited exposure to biosimilars, and even in insulin, there's a need for cheaper options. And one of the payers is saying that if the US market is not able to produce movement to the biosimilars and generate a return for those manufacturers, and then this actually can create an issue in terms of United States missing a very significant opportunity. When we look at the European biosimilar landscape, there are more than 60 launched biosimilars and multiple molecules and competing products with a diversified market across Europe. from Iceland to Turkey, from Belarus to Spain. You can see Europe is varied and a tougher to defend market, but a very interesting market. And you know, the originators mostly compete on price to share the market, and then they also play to strengthen in other areas such as education and support, logistics and inventory management, delivery device and administration mode, and environmental impact of production, and focusing on using of savings for biosimilars. When we look at Humira as an example, the first adalimumab biosimilars launched in Europe, all competing for a piece of at least 4.4 billion European revenue. These 4 biosimilars are launched at the same time, including Sandoz, Hyrimoz, Samsung, Bioepis, Biogen's Imraldi, and Amgen's Amjevita, and Mylan Fujifilm's Kyowa's Kyrin's Hulio. And there are more launching as well. So how will AbbVie defend its brand? And how will the company be competing on price, including other strategies? And what I see is biosimilars will take their place, even in the US, and it will be driven by the European markets. Some at the starting line, but may not stay the course, but big pharma's presence in biosimilars could bring credibility to the market. And the speed and the magnitude of price and market share erosion may be less painful for originators than some originally thought. But eventually, biosimilar uptake is needed if global healthcare systems are going to be able to afford pharma's next wave of innovative launches. And on behalf of my team in Ankara, Istanbul, Brussels, and Washington, D.C., I want to thank you. And again, this is Dr. Yavuz Selim Salai as an innovation management executive with our TV channels, broadcasting to 900 million people across 42 countries in Africa. And also, we are honored to support the innovative ecosystem with our VC funds, our Global Angel Investment Group, and crowdfunding platforms. And similarly, we are honored to work with entrepreneurs with great ideas and support them in their growth. And I want to thank you again for this opportunity and inviting you all to Turkey and see the booming ecosystem of innovation in healthcare and finance in Istanbul. With that, I'll stop here, and if there are any questions, happy to answer. And if not, wishing you all a great day. Thank you so much. You have all a wonderful day. Thanks a lot.
Thank you, Dr. Yavuz, for your presentation. Does anyone having any queries? I hope there are no queries. Once again, Dr.
Just one
Thank you
question,
for
please.
Okay, sorry. Okay, you can
Yeah,
ask.
just a short question. As I would like to know the opinion of you about the oral administration of biologics drugs, because until now, so we have biologic drugs which are administered parenteral. Do you think that we will have on the future oral administration?
Absolutely, definitely. There will be oral biologics, even though it's challenging. I think we will be able to see oral biologics in the midterm because there is clear opportunity for biologics. And it is just a matter of doing the right combination and doing the right strategy. And with the right strategy, we will be able to see oral biologics coming to life because at the end, if you can go around the passing route and make sure you create the right strategies for reaching to the effective areas of the body, definitely oral biologics will be in the future available.
Thank you.
And it's a great question. Thank you, Yunus. Thanks a lot.
Thanks.
And you're all welcome to reach me anytime. You can type my name on Google or LinkedIn. Just write Yavuz Salai. I'm 7/24 available through LinkedIn as well. And happy to connect with all of you. We have a great panel here. Clara, John, Judith, Ines, Imran, Shirley, Dr. Madhu, Marie, Thomas, Sophia, and Yunus and Florian. We're happy to connect with all of you and honored to host you also in New Jersey as well as in Istanbul. When you have a chance, just let me know and we'll be happy to host you. Thank you so much.
Thank you, Dr. Yamos. Now let's move on to the next presentation. The next presenter is Dr. Jonas Carrot from University of Ille, France. Hello, Dr. Jonas. Are you ready with your presentation?
Yeah, sure. Can I share the screen,
Yeah.
please?
Now I'm making you as a presenter. Kindly click on the screen button to share your screen.
Do you see my presentation?
Yes, your screen is visible now. Can you make it in a presentation mode and start your presentation?
Is it okay now?
Yeah,
I
it's
start?
perfect. You can.
Okay,
Yeah, it's
perfect.
perfect. You can start.
Thank you very much. Thank you for the kind introduction, ladies and gentlemen. It is my great pleasure to present to you our work on innovative colonic drug delivery systems based on bacteria-sensitive polysaccharide. So I would like also to thank all members of the organizing committee for accepting for this invitation. So it is a major challenge and to develop appropriate pharmaceutical dosage forms for the treatment of inflammatory bowel diseases such Crohn's disease and ulcerative colitis in order to target the distal part of GIT, the most inflamed area in Crohn's disease and ulcerative colitis. Conventional dosage form cannot be used once administered. So the entire drug dosage is absorbed and distributed throughout the human body. On one hand, serious side effects can be considerable. On the other hand, the drug concentration at the site of the action is too low, thus potential therapeutic effects are decreased. So how can we overcome this problem? So you can entrap the drug, as you can see here in red. You can entrap drug using polymeric film coatings, and an ideal dosage form should effectively suppress drug release in the upper GAT in order to prevent drug in this area, in the stomach and small intestine, but once the colon is reached, drug release should sit on and be time controlled. In the case of inflammatory bowel disease treatments, the drug is thus released and its target site providing optimal therapeutic effects and minimized undesired side effects. There are different approaches which have been described in the literature for such site-specific drug delivery. As you can see here, so you can use drug delivery systems sensitive to the pH level in the gastrointestinal tract, and these polymers are insoluble in the upper GIT, but once they get into the small intestine, or once the pH level rises, starts to be dissolved. And we can also utilize time control drug delivery systems, which are insoluble in the GIT, but become soluble in the distal part of GIT due to swelling or erosion. So another approaches are coatings with biodegradable polysaccharides, which are degraded by bacteria enzymes present in the distal part of the GIT. We find typically very high amounts of bacteria able to degrade complex carbohydrate. However, all these approaches could behave very differently as expected, because the pathophysiological conditions and the disease state may change, may vary than in the healthy subjects and might be very different from the mucosa of healthy subjects also. The pH and the GIT segments may significantly vary intra and inter individually, and also the transit times as well as the quality and quantity of the microflora, as of the microflora, which could this great care must be taken when developing such site-specific drug delivery systems for IBD patients. Otherwise, the system or the treatment will fail. So, what should be the approach for these film coatings? So, the goal of this study is to identify novel polymeric film coatings which are adapted to IBD or which are adapted to the pathophysiological conditions of IBD patients. The permeability of polymeric systems for a drug strongly depends on the drug on the water content or water uptake and dry mass loss, the weight loss. The macromolecules mobility significantly increases, and thus the free volume available for diffusion of the drug, for diffusion of the drug. The water uptake and dry mass loss of such films should be low in the stomach as well as in the small intestine in order to protect drug in the upper GIT. But once the dosage form gets into the colon, should be these systems should be permeable for drug and releasing the active agent at the site of action, as you can see here on the bottom on the diagram. So the investigated systems are, in this case, thin polymeric films, just for the preliminary studies. As a thin polymeric film will simulate exactly the composition of the film coating on the real dosage form. As you can see here, so we have manufactured thin polymeric films by casting method. So drug can be included also, and the polymer mixture, and the polymer blends are composed on ethylcellulose, which is insoluble polymer and hinders the premature film dissolution, and the second polysaccharide, which allows for colon targeting. You can so for the characterization, physicochemical characterization, you can use thin polymeric films as well as you can also ground the films into microparticles. So for microbiological analysis, we have used fresh fecal samples of inflammatory bowel disease patients, also in order to be closer to the pathophysiological conditions. And we have counted also the bacteria, as you can see here. So in IBD patients, we have less bacteria than in healthy subjects. This is very important, but this concentration is sufficient for the degradation of the film coating. So now I would like to present to you some results from thin polymeric films. And as you can see here on the left-hand side, we have characterized, or we have measured the water uptake, the water content of thin polymeric films, and on the right-hand side, the weight loss of thin polymeric films. The water uptake and dry mass loss of such thin polymeric films show a large spectrum of physicochemical properties. The ideal film coatings should show low water uptake and low dry mass loss in the upper GIT. But once the dosage form or the thin polymeric films arrives into the colon should show high water content and high dry mass loss in order to trigger the drug release at the site of the action in the distal part of G80. So upon exposure to fecal samples, as you can see here on the left-hand side, we measure the water content of thin polymeric films upon exposure to fresh fecal samples of Crohn's disease patients and ulcerative colitis patients. And on the right-hand side, you can see the dry mass loss or the weight loss of thin polymeric films upon exposure to fresh fecal samples of IBD patients. As you can see, the large property of or large physicochemical properties of these films, and the results are very promising as of for colon targeting. So, and now I can show to you the microflora identified or counted also after the incubation, after exposure, after the exposure with the without film, also before incubation with films, with the only ethylcellulose films, because ethylcellulose is not degraded by bacteria. And on the right hand side, you can see also the multiplication of bacteria upon exposure with a mixture of polysaccharide, which is degraded by bacterial enzymes present in the distal part of GIT, nutriose, and which allows for colon targeting, and mixed with ethylcellulose. It is evident, it's clearly that the multiplication of microflora is very pronounced, with films based on nutriose and ethylcellulose. This can be attributed to the fact that nutriose serves as a substrate for colonic bacteria by IBD patients. So now if we move to real dosage forms, used in this study are spherical beads, pellets, which have a dimension of 1 millimeter. Pellets are manufactured by extrusion spheronization, as you can see here on the figure, and afterward coated in fluidized bed coater with a mixer, the same composition ratio, and at different coating level, and the same composition as thin polymeric films. So now I can show you some results obtained with coated pellets with nutriose ethylcellulose at ratio 1:4 and at a different coating level. So the diagram illustrates the in vitro drug release of 5-aminosalicylic acid in percent, and in percent on the y-axis you can see the percentage of 5-ASA release. On the x-axis you can see the time of the incubation with and without digestive enzymes. So this diagram shows only the drug release and the upper GAT. And the upper GAT, that means upon exposure to simulated gastric fluid and for 2 hours, and subsequent complete medium change to simulated intestinal fluid. Solid curves indicate the release without digestive enzymes, and dotted lines or dotted curves indicate drug release in presence of enzymes, of digestive enzymes. Digestive enzymes used in this study are pepsin and pancreating. So clearly, drug release decreased with increasing coating level, as you can see here on the figure. And this is due to the increased density density of the polymer or of the polymeric network, and also the increased diffusion pathways. So we are able to almost suppress completely a drug release and the upper GIT. And this is really the ideal case for colon targeting, because you need formulation which are able to protect, to prevent drug and the upper GIT. And but this composition of these dosage form, newly developed dosage form should once gets into the colon, should increase on delivered drug in time controlled manner. So very promising results. So we have selected two candidates, nutriose ethylcellulose at ratio 1:4 and nutriose ethylcellulose at ratio 1:5. So once the colon is reached, so the polymeric film coating should become permeable and for the drug, and permeable and release the drug in time controlled manner. The diagram here shows in vitro drug release of 5-aminosalicylic acid. 5-aminosalicylic acid is used and the standard therapy of IBD patients, as is a drug which is anti-inflammatory effect, which have an anti-inflammatory effect. So, and is also a PEPA gamma agonist, so a 5-aminosalicylic acid. So we have selected two ratios, 1:4 and 1:5, at coating level of 15 and 20%. For reasons of comparison, we have studied also commercially available products used or used for the treatment of IBD, for IBD, inflammatory bowel diseases, such as Pentasa, Azacol, and Lialda. Pentasa is a drug which is coated, pellets which are coated with the ethylcellulose alone, and as you can see here, shows a premature drug release, almost 50-60% of drug in the upper GIT during 2 hours. And Azacol also is a pH sensitive drug delivery system, so which releases almost 60% in the small intestine. But Lialda shows a low drug release here, but is independent on the presence, independent on bacterial enzymes present in the colon. But very important, as our newly developed systems here, nutriose and ethylcellulose, and are able to protect drug in the upper GIT, as you can see here. So approximately 5 to 10% drug is released by diffusion, but once the dosage form gets into the colon or came in contact with fresh fecal samples, they show a sudden increase in drug release, and this is very highly promising results for colon targeting. So selected systems, also in vitro, also which show very promising results, are studied in vivo in order to confirm the proof of concept in vivo by V-star rat. And as you can see here on the figure, the in vivo study design. So the aim of this work was to evaluate the in vivo efficacy of these very promising tools to target inflammation in experimental model of colitis. In this case, also experimental model of colitis in by rats. As you can see in the representative schema of the in vivo design or study design, so male V-star rats were used, 2 control groups, the positive control groups, and the negative control groups, also with non-treated rats, but the inflammation is induced by intrarectally with TNBS, also trinitrobenzoic sulfonic acid, and the negative control intrarectally just 50% of ethanol, and the other groups received food mix technique, so without the positive control and negative control without the dosage forms. But the groups we have selected two mixture of or two coating newly developed systems, the nutriose ethylcellulose coated pellets, and also the pentasa, the commercially available product. And so in order to compare also our newly developed system with the commercially available study. So we have the blood samples were analyzed to determine the concentration of N-acetyl 5-aminosalicylic acid in blood plasma. This N-acetyl 5-aminosalicylic acid is a good indicator for the premature drug release. Why? Because 5-aminosalicylic acid, the anti-inflammatory agent, when released in the upper GIT, in the stomach or intestine, will be rapidly absorbed and acetylated as quickly in plasma. So if we have a high amount of N-acetyl 5-aminosalicylic acid, that means that dosage form shows a premature drug release. But it's a very important, as you can see here on the y-axis, the concentration of N-acetyl 5-aminosalicylic acid. On the x-axis, you can see the different formulations, and the pentasa is the commercially available product. Nutriose ethylcellulose pellets and P-starch is the two candidates or two newly developed formulations. And the vehicle only non-treated colitis does or do not show any N-acetyl 5-aminosalicylic acid. So the importantly, so rats treated with the nutriose and P-starch show significantly less N-acetyl 5-aminosalicylic acid in the blood than pentasa. So, which can be attributed to the fact that the new formulation can prevent drug 5-aminosalicylic acid in the upper GIT. So, and now I can show you some macroscopic appearance of the colon. As you can see here, so the colon of each rat was examined to evaluate the extension of the inflammation, the extension or the macroscopic lesions. Negative control, positive control, pentasa, the commercially available product, and nutriose ethylcellulose coated pellets. So the macroscopic lesions according to the Wallace criteria, as you can see here on the right-hand side, and on the right-hand side, you can see here on the right-hand side the Wallace score, and you have on the x-axis the positive control, pentasa, and nutriose ethylcellulose. Very importantly, so clearly that the inflammation disappeared as in the group of nutriose ethylcellulose coated pellets. Unfortunately, in the group of pentasa, so show an inflammation, and it's approximately similar to the positive control. So we have shown here that highly promising results with the newly developed systems, and we have confirmed also all these data with the AMEO score. As you can see here, the representative histological sections of the colon tissue, and a negative control, positive control, and nucleose ethylcellulose coated pellets, pentasa. The negative control, you can see here intact mucosa, submucosa, and masseter layer. The positive control, you have here an infiltration, necrosis, and fortunately, so nucleose ethylcellulose coated pellets, the newly developed system, show closer to negative control as data or intact mucosa, submucosa, and masseter layer. And unfortunately, the pentasa shows the infiltration into the submucosa and also the mucosa is disappeared. This is very good information of the in vivo efficacy of the novel coating. So we have also measured colonic MPO activity. Also colonic, the MPO is a marker of inflammatory cells that participate to the TNBS-induced colitis. So figure shows the concentration figure shows the concentration of colonic MPO activity. On the x-axis, you can see the different formulations, the vehicle only non-treated colitis, the group of non-treated colitis, which show high concentration of MPO and the tissue on the colon tissue. The pentasa is the same, unfortunately, but fortunately, so nucleose, ethylcellulose pellets, and pea starch, so the two newly developed systems show less concentration of MPO in the tissue of the colon. Again, this is as most promising results shows with nucleose and pea starch. We have also measured the pro-inflammatory cytokines which are involved in the inflammation, so interleukin-1 beta and TNF alpha. And again, here you can see the concentration of interleukin-1 beta is less pronounced than pentasa and of course than non-treated colitis. The pro-inflammatory mediators, as I said, are involved in the inflammation and also in the disorders of the inflammation, of the dysfunctions of the human intestinal epithelial cells. So, and also which play an important role in the severity of the inflammation. So as I said at the beginning of the presentation, 5-aminosalicylic acid is an anti-inflammatory drug, which is an agonist of PEPA gamma receptors expressed in the colon and the distal part of GIT, and we have quantified in the tissue, on the colon tissue, the PEPA gamma receptors. So the PEPA gamma receptors, if you have an activation, are down-regulated and the inflammation or and the disease state down-regulated. And if you show high concentration of the PEPA gamma receptors and its target genes, in this case hydroxymethyl glutaryl CoA synthase, so that means that the inflammation would would be ameliorated using 5-aminosalicylic acid. And as you can see here on the figure, so on the x-axis you can see the different formulations, and the y-axis the concentration of the target genes of the PEPA gamma, and you can see here that the concentration is more pronounced. It's very pronounced, more pronounced than in group rats treated with pentasa, as well as in rats non-treated or vehicle only. This is very promising, and will confirm really the proof of concept in vivo by rats. So we have analyzed, or we have measured the activation of PEPA gamma receptors in transgenic mice. And the peroxisome PEPA gamma is the peroxisome proliferator activated receptor, and has been identified as a key regulator and new therapeutic target in inflammatory bowel diseases. The aim of this part of this study is was to demonstrate the activity of PEPA gamma or of such receptor in ex vivo administering drugs, such drugs or administering the newly developed systems nutriose ethylcellulose pellets, as well as the commercially available product pentasa, as you can see here on the bottom. So on the top you have the transgenic mice treated with nutriose ethylcellulose pellets, and the bottom treated with pentasa. So the pentasa shows all experiments were carried out with the 7 week mice of 7-week-old male PPU Luc mice, and the bioluminescence reporter of the activity are quantified, and we have quantified the photons, the emission of the photons, and you can see here that and the group of nutriose ethylcellulose pellets, the bioluminescence is very pronounced, more pronounced than in pentasa group, and the intensity is very pronounced during 4 days or 4 days. This is the time, the period of the treatment, and unfortunately here, so the pentasa, the bioluminescence is decreased during this period. This can be attributed simply to the fact that our system, the nutriose ethylcellulose pellets, protect drug in the upper GIT. However, pentasa releases drug, almost 60% of drug, or 60% of drug in the upper GIT. Drug is absorbed in the whole or distributed in the whole, in the entire in the whole body, and the concentration which arrives in at the site of the action is very low in case of pentasa. So to conclude, we have identified very promising novel film coatings for colon targeting, which are adapted to the disease state. This is very important, and we have shown, or we have shown negligible drug release and simulated gastric fluid and intestinal fluid, so we are able to protect drug approximately, so almost 100 or 90% of drug in the upper GIT. The system are sensitive or susceptible to bacterial enzymes present in the colon in the colon of IBD patients, and also we have shown the sudden increase in the drug release upon exposure to fecal samples of IBD patients. This is very important. On all these data show confirm the superiority of the the proof of concept in vivo and the superiority of the newly developed systems in colon targeting or for the treatment of IBD patients. Thank you very much for the organizing committee again for this invitation, and thank you very much also for your attention.
Thank you, Doctor, for your presentation. Does anyone having any queries? I hope there are no queries. Once again, thank you, Doctor, for your presence and presentation.
Thank you very much.
Now, as per the schedule, now let's take a lunch break and we'll get back with the afternoon session. Stay connected.
This conference will now be recorded.
Now, let's continue our webinar with the afternoon session. The first presenter is Dr. Maria Sanatia from Orabashko Thermonil National Medical University of Thermonil, Ukraine. Hello, Dr. Maria. Are you ready with your presentation?
Hello, yes.
Okay, Doctor, now I'm making you as a presenter.
I will share my screen, of course, yes.
Okay, now I made you as a presenter. Kindly click on the share button to share your screen. I have made you as a presenter.
Yes. Do you see my presentation?
Yeah, it is visible now. Can you make it in your presentation mode and start your presentation?
Okay, thank you. Dear organizers and participants of the conference, thank you a lot for this possibility to take a part in such prestigious conference. Greetings from Ukraine. As it is known, the medicinal plants of the Lamiaceae family of the Nepidoideae family of the Lamiaceae family are very famous in the pharmaceutical markets of Ukraine and throughout the world, because there we have lemon balm, herb, or menta piperita, peppermint, sage, thyme, oregano, etc. The use of alternative sources of essential oils, as well as extracts and comprehensive pharmacognostic study of unofficial plant species from this family were the aim of our study. When we decided to analyze the presenting of representatives of the other genera of this family in the pharmaceutical markets of our country, we found that only several species are presented. For example, Otimum sanctum, or Hyssopus officinalis, or Saturea hortensis. So the main task of our study was to conduct the complex pharmacognostic analysis of many species, of the family Lamiaceae, to provide the, I cannot change the slide. Lamiaceae family, we put the aim to establish the content and presence of main group of biologically active compounds, mainly of the secondary metabolites. Next point was to carry out the macro and microscopic analysis of raw plant materials of these representatives. On the next stage, we developed the technology of obtaining new phytomedication on the basis on raw materials of the studied species and to detect the parameters of their standardization. And at the last stage, we conducted the pharmacological studies of the obtained phytomedication in vivo and in the in vitro studies. The first objects of our research were 4 species of the Otimum or basil genus, Otimum americanum, Otimum basilicum, Citriodora, and Sanctum. Next one was the Hyssopus officinalis from the Hyssopus genus. Next objects were Monarda fistulosa, Saturea hortensis, Lophanthus anisatus, Dracocephalium grandiflora, and Dracocephalium moldavica. It should be mentioned that we used 2 forms of Lophanthus anisatus and Dracocephalium moldavica. Their difference were in the color of the flowers, white and violet forms. The plants were grown in the Ternopil region, Ukraine. The aerial parts of the plants were gathered at the beginning of flowering period in summer. The herbs were dried at the appropriate temperature, and the raw materials were sieved through sieves of the appropriate diameter as specified in the relevant research methods. In the initial stage of research, we do the phytochemical analysis. We did this phytochemical analysis of the essential oils separated from the aerial parts of plants using hydrodistillation method. Later, we use the chromatography, gas chromatography mass spectrometry method was used for the chromatographic analysis of essential oils. For example, we found that linalool was the predominant compound of Otimum americanum essential oils. Carvacrol and hamaterpinen were the major constituents of Saturea hortensis essential oil. All these plants we analyzed through gas chromatographic analysis and found that 5 of the previously studied 12 representatives were the most promising for further pharmacognostical analysis. For example, we have chosen the Dracocephalium moldavica, Lophanthus anisatus, Monarda fistulosa, Otimum americanum, and Saturea hortensis essential oils. We found that the main biologically active compounds and chemically significant compounds of these essential oils, we can analyze them using also fast method like thin layer chromatography. On the next stage, we did the HPTLC analysis of polyphenols in the herbs of all studied species. We conducted these studies in Poland at the basis of Opole University and found that rosmarinic acid was the main biologically active polyphenol of all 5 studied species. we use several standards of polyphenols, and only 6 of them were found in different of studied species. For example, apigenin, luteolin, rutin, caffeic acid, rosmarinic acid, and chlorogenic acid. As it was mentioned, rosmarinic acid was the main biologically active polyphenol of all studied species. On the next stage, we detected the rosmarinic acid in the 5 species using HPLC densitometry, and we found the quantity. Quantitically, the amount of this hydrocinamic acid. For example, the biggest content we found in Dracocephalium moldavica, and the minimum content was in the Lophanthus anisatus herb. On the next stage of our research, we investigated the polyphenols contents, rosmarinic acid, as well as other hydrocinamic acid or flavonoids using a high-performance liquid chromatography method. And as can be seen from the result, Dracocephalium moldavica also contain the biggest amount of rosmarinic acid. The contents of polyphenols determined by HPLC method we can see from this slide. Mostly among flavonoids, apigenin and luteolin glucosides dominated in all the species. Among hydrocinamic acids, rosmarinic acids, and sometimes ferulic, for example, in Monarda fistulosa, or caffeic in different species, were present in the significant amount also. Then we studied the total contents of hydrocinamic acids spectrophotometrically. We compared the light absorption spectrum of different hydroxycinamic acids and our extracts, and found that the maximum of light absorption was at about 505 nanometers after addition of nitrate molybdenum reagent. So after comparing these maximums, we did the research, spectrophotometrical research at the wavelengths of 905 nanometers. As it can be seen from the slide, Monarda fistulosa and Dracocephalium moldavica accumulated the most significant amounts of hydrocinamic acids totally. After analyzing different results, we conducted that Monarda fistulosa herb is the most interesting for further deep study. And we use dried herb of this species for isolation and detailed analysis of different polyphenols. On the first study, we did the maceration with 70% ethanol using periodical shaking. After it, we did evaporation of the extract down to the water residue, and then we conducted the liquid-liquid extraction with chloroform, ethyl acetate, and butanol. TLC analysis and separation of fraction, column chromatography, preparative TLC chromatography, and then we use crystallization and recrystallization. After this sort of investigation, we conducted the HPLC analysis of different fractions from the Monarda fistulosa herb. On the slide, it can be seen the HPLC chromatogram of chloroform fraction at daylight, where the standard of thymol we used. Next, butanol and ethyl acetate fractions on the middle figure. And lastly, we use the water fractions. The chromatographic separations of fractions we did using gradient elution of fractions in a glass column. Then we collected some subfractions and analyzed through HPLC chromatogram. For example, on the slide in bottom, you can see the butanol subfraction of and different chromatogram tracks. Finally, 6 individual compounds were isolated from the Monarda fistulosa herb, and their structure were proved by spectroscopy, mass spectrometry, UV spectrophotometry analysis. Later, we analyzed the microscopic and microscopic features of the studied plants. For example, on the slide, I put the specific microscopic and microscopic features of Monarda fistulosa herb. It has specific rectangular stem with rounded ribs. If we can see from the next slide, for example, Otium americanum herb, its stem is very specific with protruding ribs. And we can analyze each organ, and there we can see many differences in the structure of leaves or upper and lower epidermis, trichomes like simple hairs or glandules or glandulous hairs. So we have proven the specific diagnostic features according to morphological and anatomical structures of all 5 studied species. Next point, we developed the general technological scheme for preparing different phytomedications from all plants. For example, we prepared the dry extract from the Monarda fistulosa herb by complex processing of raw material. It means that on the first stage, we obtain essential oil by the hydrodistillation method. And after this, we conducted the development of dry extract from the post-distillation raw material. Next, we studied through chromatographic analysis using high-performance thin layer chromatography or high-performance liquid chromatography. And we found the most significant chemical compounds of each preparation. The results of these studies of the Monarda fistulosa herb were published in 2021 in a prestigious scientific journal, Frontiers in Pharmacology. We also analyzed and found many significant features of Otium americanum tincture and different extract, essential oils, infusions. These results also were published last year. the spontaneous locomotor activities of experimental animals we studied during the in vivo research of sedative effect of different studied substances. As it should be mentioned, the lemiaceae plants are used for sedative effect often. For example, Melissa officinalis or Thymus serpyllum or Mentha piperita. So we continue this study according to analyzing the the other lemiaceae species and found that Otimum americanum herb as well as Dracocephalium moldavica herb are the most important and the most appropriate for preparing the sedative drugs. On the next stage of our studies, we conducted the comparing analysis of the antimicrobial activities of essential oils of 5 species. And we found that Dracocephalium moldavica as well as the other species were mostly the most effective against gram-positive microorganisms. Lophanthus anisatus was very effective against Candida albicans, so it has antifungal activity. We determined this antimicrobial activity using the Wells method in concentration of essential oils 10 milliliters milligram per milliliter. And the next stage, and we dissolved our essential oils in the dimethyl sulfur oxide. The Staphylococcus aureus and Staphylococcus aureus was mostly inhibited by the Lophanthus anisatus and Monarda fistulosa essential oils. We also studied the antioxidant activity of infusions prepared from the herbs of the studied species. We use the in vitro method using the DPPH radical. The most significant antioxidant activity was found for the Dracocephalium moldavica herb. And we found the positive correlations between the content of polyphenols in the studied infusions and their antioxidant activity. Thus, the comprehensive phytochemical analysis of several unofficial medicinal plants of the Lemiaceae family has shown that 5 species such as Monarda fistulosa, Lophanthus anisatus, Saturea hortensis, Otimum americanum, and Dracocephalium moldavica are the most promising for further pharmacological studies. The standardization parameters for the raw materials and obtained medicinal products were established. The analysis of the depends of biological activity of the developed herbal medicinal products on presence and contents of their main biologically active compounds were concluded. The results of these studies were published recently in the different journals from the Scopus database. Thank you a lot for your attention, and many thanks for the support provided by many countries in my country's defense against the Russian aggression, which we feel nowadays. Thank you.
Thank you, Dr. Maria. Does anyone having any queries?
Maybe one short question about the products you have selected. Is it for a local treatment, or could be also imagined for oral administration? Or
Thank you. We suppose we conducted the in vitro and in vivo studies, yes, and we didn't provided clinical studies nowadays. We suppose, for example, that tinctures and essential oils we can put in future, for example, in elixirs or sprays for the oral cavity. Or in gynecology, we are planning these studies in future, and the present stage we studied only in vitro and in vivo. Sorry.
because does it exist or already solution based on Lamiace for local treatment of the herpes virus?
Herpes, okay, I understand your questions. Nowadays, we don't study antiviruses activity,
Okay.
yes? We are study antibacterial, antifungal, viruses. I hope in future we could conduct this very important research.
Okay, thank you very much.
Not at all, thank you also.
Thank you, Dr. Yunus. Does anyone having any more queries? I hope there are no queries. Once again, thank you, Dr. Maria, for your presence and presentation. Now let's move on to the next presentation. The next presenter is Dr. Madhu Gupta from Delhi Pharmaceutical Science and Research University, India. Hello, Dr. Madhu. Are you ready with your presentation?
Yes, doctor. I am ready. So
Okay,
please give
Dr.
me
Madhu,
option
I'm
for.
making you as a presenter. Kindly click on the screen button to share your screen.
Yes.
Your screen is visible now. Can you make it in a presentation mode and start your presentation?
But first slide is missing here. I just try one more time. Okay.
If you have another copy, can you open that?
It's okay. I think there is some technical issue, so no issue. I just go to presentation mode.
Okay, please make it in a presentation mode and start your presentation.
Yes. So first of all, a very good afternoon to one and all. This side Dr. Madhu Gupta, and today I am presenting a topic that is the is placental therapy. It is a significant tool for wound healing. So first of all, I go to details with there is some context. So according to the Indian context or International Diabetes Federations, am I audible? I think.
Yes, you're audible. You can
So
start.
there is an increasing pupils for diabetes case, and if we talk about India, so more than 25% of the total diabetic population suffers with diabetic foot ulcer, and all we have know that every 20 seconds one amputation was there. However, there are so many marketed formulations are available in the form of various drug formulations, that is wound dressing, debridement, bioengineered products, some hyperbaric oxygen therapy. But however, there is some issues. So the if we discuss about the customers, so diabetologists, endocrinologists, general physicians, these may be the possible targets for particular disease, particular formulations. If we discuss the emotional impact for case of wound healing therapy, so patient-centric, poor quality of life, depressive and anxiety symptoms. These are the basic emotional impact that will exist in the patients. And there are some alternative shortcomings, that is the accessibility and affordability, chances of remission, stability issue, and biofilm formation. So we have after all these things, we decided to go for the wound healing based formulation. So we also analyze the market survey, and it will increase around 6.2%. And there is increasing the prevalence rate of chronic disease like obesity or diabetes. The It will increase in the adult and geriatric populations. There is some technological advancement in formulation, and some traditional formulation that is having or having the some shortcoming. So all these factors push up for development of some innovative wound healing formulations. And as we know, impaired wound healing, or we can say the chronic wound healing, it will take around more than 20 weeks for complete healing. And but chances of remission, it is very frequent, and the resistant biofilm formation. So all these factors we have considered, and we have developed the safe and efficient products. And we already this concept was established at preclinical level. Now we are planning for move forward and some pilot plant study. I am not able to move the slide actually. Ma'am, I am not able to move the slide. Hello? Yes, ma'am, I am not able to move the slide.
Can you please change one?
Yes. So the
Yeah, it
proposed
is.
solution. Yes, thank you. So the proposed solution in terms of technical steps. First, we have processed, identified, and characterized the placental extract, that is the animal-based placental extract, and we have termed as regenerate. And we have checked its safety and toxicity profile. Then we turned this placental extract into the gel, that is patient compliance, and we have measured its therapeutic efficacy at preclinical level. So for first, the raw material is the placental extract. So we have characterized this placental extract, and it provides the maximum number of peaks. And this characterization was done on the uh, GPC and mass chromatography, and we observed that maximum number of peaks was observed in the 6,000 to 6,800 Daltons. And this placental extract, this is basically the biological active constituent that is rich with growth factors of cytokines, growth factors, and some other regenerative based important constituent. And this regenerative effect of this placental extract, and this is impact on the signaling mechanism of intracellular interaction. So we have visualized all these concepts. So what is the novelty for this formulation? This is the first scientifically proved report that anyone have used this placental extract for treatment of particularly wound healing. And this formulation is we have checked its preclinical level. So we have found that it's reduced the wound healing time. It provides a unique skin regeneration property, and most important, it can also able to really that is the in bio wound healing therapy, biofilm formation is very frequent, so it is able to overcome that type of problems. And more important, it also provides the sustain and control regime. If we Yes, we have also granted this patent. We have filed the patent, and we have granted in this year itself in Indian. And this raw material was provided by the Russian Medical University. So I think if there is some issue. So now there is some biological products available in the international market and Indian market, but they have some side effects. They show some, however, they show some beneficial effect, but they also show some side effects, such as shorter half life, causes a rash and burning issue, and they are more costly. Patient are not able to sustain that. Patient cannot afford this. So these all problems are exist. But in case of, yes, Indian market is also, in Indian market there are some formulation are also available, that is the biological based, but they also have some shortcomings like skin irritation, rashes, pain at the site of infections, rectogenicity, allergy. So we have proposed a regeneration based solution. It provides the wound healing by tissue regeneration and provide the minimal scars. It reduces the healing time. It's provide the anti-inflammatory and very good antioxidant property. It is having the unique skin regeneration property. So it will ultimately affect on the wound healing. How this formulation is differed from the proposed solution. This is the first novel topical gel formulation, and it has showed the resistant against microorganisms and biofilm formulations. And this novel design formulation can potentially overcome the challenges that is associated with the market formulations. And this formulation can be easily formulated in the simple setup of lab, means we don't require so many higher infrastructure. Yes, everybody know what is the wound, so I'm not going to detail of the wounds. Generally, wounds are classified according to tissue loss, but it is very basic. So wound healing process, it involves the four process. Wound healing, that is the hemostasis, inflammatory, proliferative, and remodeling. These four phases of wound healing are exist. So now the diabetic foot ulcer, we have checked the therapeutic efficacy of our formulation in the diabetic foot ulcer based chronic wound. So we all know the epidemiology of diabetic foot ulcers, how the this type of indications can affect the patient's life, quality of life, and it is very costly. So every person cannot afford this type of higher costly treatments. So this is the epidemiology and financial burden of DFU. So what is the need of newer approach? We all know that in every 20 seconds there is one amputation. So all the available formulation, they are having very increasing risk of safety, means and they are very costly. So every person cannot afford. Plus these formulations are provide the effect, but the duration of treatment is very higher, means it will require a longer duration of time. So we are having a solutions that is able to reduce a shorter time plus provide the unique regeneration property. So we know, yes, is regenerate or placental based extract gel, it might be a perfect solution for wound healing therapy. So as all we know, this is a placental extract. This is having a very important different types of proteins, that is composition in the placenta, including the growth factors, hormones, cytochromes, and fibrinolysis factor. These all are very important for the wound healing. And all we have know what is the role of growth factors in the wound healing. So we have termed this animal placental extract to the regenerin, and this regenerin, we come to know in this regenerin mostly the 20 to 200 amino acid based residue that is important for this wound healing therapy. So we have done this characterization by the mass spectroscopy, as I already discussed in the previous slide. So this is the result interpretation. We have identified about 30 signals of low intensity, and the range is 3000 to 333 Dalton, as well as most of the peaks are observed at 6000 to 6800 Dalton, and the most intense signal that we have received, that is a 6303. So we have considered the growth factors or the energy providing bioactives that is in the comes under this region. So yes, we also characterized our prototype of drug by with this chromatograms that these show here, and these are the peaks height that we have observed in different time periods. So molecular composition of regenerate data of mass spectroscopy analysis and size exclusion chromatography. All these characterization parameters, the results supported that the sample peptides with a low molecular weight around 6,000 Dalton, and it indicates the presence of valuable biological active components, growth factors, cytokines, and cytamines. These are responsible for regeneration and proliferation of skin cells. The composition of the peptides on the basis of light fractions and heavy fractions. So these are having the some growth factors that is responsible for the regeneration property. For example, growth factors, insulin-like growth factors, epidermal growth factor, and transforming growth factors, VEGF. So these are the very important bioactive constituents. Those are present on the placental extract based gel. Placental extract, actually we have turned this placental extract into the gel. So why we taking further this formulation from placenta region? Because it is having super antioxidant, it is having antimicrobial property, it is having anti-inflammatory, it provides the cellular proliferation, tissue regeneration, and it also provides the efficacy in terms of wound healing. Some literature also reported that this type of placental extract can be used for alopecia or psoriasis cases. So, as I already discussed, what is the composition of these elements? So, this is the whole experimental methodology, how we perform this our experimental setup. So, this is the in vivo study protocol. So, it already take around 65 days to complete all the study. Before going to the in vivo study, first we have taken the placental extract, then we have turned into the gel, then we have characterized this gel with various types of evaluation parameters. So that is not contained here. So what this placental extract based gel we have topically used. So this provides a local action, reduce the side effect, it provides the sustained and controlled action, plus it improves the patient compliance. So all the studies were performed in India, and its safety and toxicity study was performed under the OECD guideline 402. And we also checked its therapeutic and safety potential of regenerating diabetic foot ulcer treated rat model. So this is the protocol. Now we have also performed its pre-formulation study. So if we take its nature is liquid, the color is transparent, and its odor is odorless. Solubility study, it is highly soluble in water soluble. UV spectroscopy is 364 nanometer. So we have not contained here the in vitro study. We directly comes into the in vivo studies. So first, we have induced the animals with diabetes, and after diabetes, we have increased the blood glucose level by giving the insulin. So by giving the glucose, so we have in this data, we have maintained this blood glucose level in the rats around 1 to 10 weeks, and we found in this table that this blood glucose data is more than 300 mg per dl, and it is maintained up to 10 weeks. If we consider about the body weight, so if we injected the streptozotocin on rats, that produced a significant decrease in body weight as compared to the control group. We have taken the 5 groups and study periods up to 1 to 10 weeks. In first group, we have taken the normal control, that is the healthy animals. G2 is the diabetic control. That means we have induced the diabetes, but we did not treat it with any type of formulation. In G3 group, we have taken the marketed formulation. G4, we have taken the low dose based regenerin gel, and G5 is the last group that we have taken the higher dose of regenerin gel. So we have found that consistently rats will is decreasing the body weight. If we consider the food consumption and water intake, so we found that yes, lesser food intake and water consumption as compared to the normal group. So this is the graph that is already represented. Yes, water intake. So water intake by rats during DFU experimental setup are also checked for different time periods. So wound size. This is the first study that we have checked the wound size. We have adopted the excision wound model, and the study duration is 1 to 18 days. After the blood glucose level is more than 300 mg per dl, then we have induced the excision wound, and then we have checked the therapeutic efficacy. So we found that standard groups showed the treat means it will take around 18 days to almost complete the contract the wound size. But in case of regenerin based gel, it shows the effective results on day 5, and up to day 15 it was almost healed. And in case of high dose, it was only 1.53%, almost no wound was there. So this higher dose, that is 1.2 mg per ml based gel, that is most effective in as compared to the all treated formulation. So we can say yes, this wound size is also based on the time dependent as well as the formulation dependent, right? Next study that we have performed is the wound closure percentage, in which we found that regenerin based gel that produced the time dependent significant wound closure in rat. As we can show that more granular tissue formation plus regenerin based gel provided that in this table you just show कि after 15 days or in 15 days there is 99% efficacy was there, or complete wound closure was there in case of higher regenerin based gel or G5, and in case of G4 it is 87%. It is also good result, but in case of marketed formulation it is around 72%. But so we can say that yes, more we can say 15 days is ideal, and almost 15 days the wound closure is almost minimal or completed. We also checked the mean epithelialization day, and we found that yes, regenerin based gel provides induced for treated formulation based treated group provide the complete epithelialization as compared to all the treated groups. This is the morphological analysis of the rats that was treated with different formulation, and we found that these last two group picture that is showing the regenerin based gel, and we found that in day 15 the minimal scar is present in case of higher gel treated, and in 18 days it is completely vanished. So we can say in both cases. So we can say the regenerin based gel it is most effective formulation, and we also checked, and this is the morphological structures. We also checked the C-reactive protein for determining the inflammatory nature. So we found that hyperglycemia delayed the wound healing with the reduced inflammation and CRP, and reduce the cell proliferation in wound. But all the treated groups as compared to the normal control group, even that condition, the reduced CRP level. This is the, we can say, in diabetic control group, this is the higher CRP value, but in case of, yes, in standard, it is although significant, but in case of regenerate gel treated based group, it is showing the low lesser CRP value. So we can say that yes, in this case, the less inflammatory reactions was there. So drug provides the anti-inflammatory nature. Yes, next slide. Yes. So we also check the hydroxyproline estimation to know or identify the recognition of the collagen deposition. So we found that its significant formulation treated group increase the wounded granulation tissue on the application on the application as compared with diabetic control, and it significantly and it is showing the effectiveness significantly as compared to the regenerative group, and it is the matured collectively acts in the healing of the wound, and this increase the concentration of a collagen tissue to and strand to the tissue for repair. We also perform the histopathology. This is the histopathological scores, and we found that the score is very less as compared to all the means G4 and G5 give the best morphological estimation, histopathological estimation, and there was complete reepithelialization. These all 5 pictures. First picture showing the normal control, second picture showing the diabetic control treated group, third picture is the standard group, 4 and 5 is the formulation treated group. So we found that there was complete reepithelialization, mature collagen fibers, neovascularization and fibroblasts and less inflammatory cells are observed in the treated group in these 2 pictures. And this histopathology study suggested that inflammation continued with the aggregation of neutrophils, that is between the layers and in the epidermis, until day 18 post-injury in the foot wounds, but in the diabetic control group. But in the case of normal control group, it subsides on the 7 days. And in case of formulation treated group, there was very less aggregation of excessive neutrophils that is responsible for inflammatory reactions. So on these basis, on all the studies analysis or all the studies results supported this, yes, this formulation was effective, and we can explore there is some issue on yes. So last slide is the conclusion. So our data, all the studies data supported that regional is a safe formulation, and we can explore for further applications. This study provides perhaps for the first time, and as per our knowledge, best of our knowledge, and it is very effective in diabetic foot ulcer. It is basically a regional is an animal model of diabetes that produces a wound contraction, epithelialization, and modulation of inflammatory mediators. So, as per our results and the study protocols, we can say yes, this is a very effective formulation, and in future we can go for explore the more study, and we can explore the more results. So thank you for everyone for patience listening. All this is all from my side. If anyone having any query, I'm happy to address the queries.
Thank you, Dr. Madhu, for your presentation. Does anyone having any queries?
I have a question. Is it rats? Is it an appropriate animal model for diabetic foot ulcer, or is it you have used rats is just because not expensive?
Sir, we have referred this model with the previous literature paper, and we have taken here is the albino rats, the star albino rats. So we have checked our whole study in this model, and we have checked our study on the excision wound based model. So yes, this model is already reported in previous papers. So we have taken as a reference for previous papers.
Okay, thank you.
Thank you, sir.
Does anyone have any more queries? I hope there are no queries. Once again, thank you, Dr. Madhu, for your presence and presentation.
Thank you.
Now we'll move on to the next presentation from Dr. Shubhan Naman from Maharaja Ranjit Singh Punjab Technical University of Mathida, India. Hello, Dr. Shubh Naman. Are you ready with your presentation? Okay, we'll move on to the next presentation from Dr. Shubin Mary Jacharia from AIMS Health Science Campus of Kochi, India. Hello, Dr. Shubin Mary. Are you ready with your presentation? Hello, Dr. Shubin. Can you please unmute
Yes,
yourself?
madam. Yes, madam. I am ready.
Okay, now I'm making you as a presenter. Kindly click on the screen button to share your screen.
Yes. Can you see?
Yes, your
Is it
screen is
okay?
visible. Kindly make it in a full screen mode and start your presentation.
Is it okay?
Yeah, it is perfect. Can you make it in a presentation mode? Yes, perfect. Now you can
Yeah.
start.
What about now? Yeah. Good evening. 1 second. I want to minimize the screen.
You can move the go to midis toggle.
Ma'am, is it ready?
Yeah, it is ready.
Yeah. Good evening, all. Myself, Dr. Subin Mary Sakriya. I am working in Amrita School of Pharmacy as Associate Professor. Today, my title of presentation is Identifying Mucormycosis Severity in Indian COVID-19 Patients. It is a study on nano-based diagnosis and its necessity for critical therapeutic intervention. So as we all know, COVID-19, it is also known as SARS-CoV-2. It is caused by coronavirus. Since the emergence of the disease, several studies have been conducted in terms of the pathophysiology, diagnosis, treatment, prognosis, complications, etc., and which was found to be unexpected and unreliable. Coming on to the clinical presentations of black fungus, which is also known as mucormycosis. The major symptoms are headache, sinusitis, blackish discoloration, local pain on the cheekbone, tooth decay, black spots in front of the eyes and pain with blackish pupil. And these are some of the pathological evidences of black fungus. The inhaled sporangiospores will go into the distal alveolar space, and the mononuclear cells and the polymorphonuclear cells will destroy the spore formation. And after that, there will be a fungal invade of the free ion leading to the growth of the hyphae. And finally, this hyphae, it will invade. It will invade, and that will result in hemorrhage and necrosis. That will lead to severe hemorrhage as well as necrosis. And finally, that will lead to the system's immune response will be highly affected, and it will go into the bloodstream. So in the final stage, there will be an extremely excess of fungal growth. Fungus will grow rapidly, and that will result in the failure of our immune system's defense mechanism, and it will reach the bloodstream. Moving on to the statistical data, there are in India, ICMR has released a lot of guidelines for the early detection and prevention of mucormycosis. And based on these guidelines advise the patients who were recovered from COVID-19. That is usually the patients who are affected with COVID-19. As we all know, the COVID-19 patients will develop this mucormycosis at the advanced stage. So ICMR has released some guidelines in order to that is for the earliest indication of in order to understand the disease as well as understanding the indications as well as clinical symptoms of the disease. So a multidisciplinary approach with the help of ophthalmic surgeons, then neurosurgeons, general surgeons, etc. were done. And they recommended the drug of choice of amphotericin B and posaconazole. And our Union Health Minister, Dr. Harshvardhan, has advised the patients to avoid self-medication, especially with steroids, because that will lead to dangerous situation, that will lead to detrimental effects of the infection. And at this and also along with that, following this, ICMR also released the guidelines for the management of the disease, especially in the case of diabetes patients, that is by controlling the diabetes, reducing the uncontrolled use of steroids and immunomodulatory diseases, etc. So there are some approaches to mucormycosis using nano, that is nano-based diagnosis. So that process involves fungal detection biosensors, diagnostic based on nucleic acid, then POCT, that means point of care test, and detection of galactomannins. And there is always an interplay between the antifungal therapy as well as COVID-19. So there are antifungal drugs are prescribed in COVID-19, so that can be used in mucormycosis. Examples for that antifungal drugs are especially novel triazoles like posaconazole, voriconazole, then isavuconazole, and echinocandins such as caspofungin, anidulafungin, etc. And many trials also highlighted the medications tocilizumab, interleukin, IL-6 receptor antagonists, etc., which can also regulate the dangerous inflammatory processes. So all the above, surgery will be the final remedy, that is a final treatment. So according to physicians, controlling the insulin, reducing the steroids, as well as stopping of the immunomodulatory drugs are very important. And in order to ensure proper, that is, in order to avoid dehydration, there's a procedure that includes an IV infusion that involves daily saline supplement is given, along with an infusion of amphotericin B. So that will that should be continued up to 1.5 months. Steroids should be used with caution, and there should be proper planning. And at the same time, in the case of especially diabetes patients, should control their hyperglycemia. They should regulate blood glucose levels, etc. Then the most unfortunate event is that there's a lack of available clinical trials existing. So that makes it difficult for the physicians to select the correct antifungal agents for treating mucormycosis. These are some of the, as we already discussed about the antifungal agents, posaconazole, voriconazole. The structures are given here, the chemical structures of caspofungin, etc. Then also the gastrointestinal tract also will be affected, followed by because of mucormycosis infection. Especially in immunocompromised patients, that will cause substantial mortality up to 54, up to 50, more than 50%. So these are some of the plausible reasons causing black fungus infection. That is, first one is improper use of steroids, then irrational use of antibiotics, then poor hygiene. So that is a very important factor that can cause mucormycosis. Then uncontrolled diabetes, that already we discussed. So these are some of the clinical, you can see these are some of the symptoms occurring in diabetic patients, that is right orbital cellulitis. Then this is the palatal mucormycosis affecting the mouth, affecting the jaw, and this is the cutaneous mucormycosis. So you can see the face here. This is the problems, right orbital cellulitis of the eye, then palatal mucormycosis, then and cutaneous mucormycosis of the skin. And coming on to the prospects and recommendations, Bajaj Healthcare has recommended amphotericin B as a for treatment for mucormycosis. And at the same time, parental posaconazole was also been considered, especially that will reduce the, it is one among the important advantageous therapy options available. Oral tablets of posaconazole that will prevent the recurrence, and mortality is expected more in patients with uncontrolled diabetes mellitus. That is a major problem. So we need to administer the correct dose, and duration of steroids should be minimized in the case of diabetes patients. So the conclusion is that mucormycosis, especially the etiopathogenesis of the mucormycosis, differ from nation to nation, and the disease appears to be very severe. So we have seen the clinical spectrum in the case of in the case of diabetes patients, uncontrolled diabetes patients. Mortality is very much high in diabetes patients. So early interventions such as surgical debridement and medicines can help to improve the status of this dangerous illness. These are the references. So it was invited. I was given this the topic because it was published earlier last year in an antibiotics journal. So I was told to talk about this subject. That is why I selected the same topic. Thank you all.
Dr. Subin, for your presentation. Does anyone having any queries? I hope there are no queries. Once again, thank you, Dr. Shubhamari, for your presentation. Now let's move on to the next presenter. The next presenter is Dr. Shubh Naman from Maharaja Ranjit Singh Punjab Technical University of Bathinda, India. Hello, Dr. Shubh Naman. Are you ready with your presentation? Hello, Dr. Shubh Naman. Can you please switch on your mic button?
Yeah, I already
Yeah.
switched on the mic button, ma'am.
Okay, now you
Hello.
are audible. Now I am making you as a presenter. Kindly click on the screen button to share your screen.
Yes, ma'am.
Hello, Dr. Shubnaman. Are you there? Hello, Dr. Shubnaman. Hello, Dr. Shubnaman. Your screen is visible now. Kindly open your PPT and start your presentation.
Hello.
Yes, Dr. Shabnaman.
My screen is visible now.
Yes, your screen is visible. Can you please open your PPT and start your presentation?
It started the PPT also.
Yeah, now it is visible. Okay, can you make it in a presentation mode and start your presentation?
Yeah, ma'am. It's visible.
Hello, Dr. Shabnaman.
Yes, ma'am. Yes, visible. My PPT is visible now.
Your PPT is visible. You can start your presentation. May I know how you're facing any issue?
Okay. Good evening. It's a very windy here. That's due to a bad network. There is a power cut also, so I'm facing this problem of internet also here. Sorry for inconvenience. Uh, I'm starting my PPT presentation on quality and design-based formulation of paracetamol
Okay,
containing
can you
physical
open your
granules and
PPT?
practical
We are
alternative.
unable to see your PPT. As you have opened your
It's
browser window,
it,
can you minimize the other windows and open your PPT, please?
it's visible, ma'am.
Your screen is visible, but your other windows are open in between. Kindly please minimize the other windows and open your PPT.
I think so. Now it is visible now.
Yeah, it's perfect now. You can start your presentation.
So sorry for inconvenience. There is a network issue due to the windy condition here. Uh, I am starting my presentation that is by quality by design based formulation of paracetamol containing fizzy granules as pediatric alternatives. Hello everyone. Myself Subnaman from Department of Pharmaceutical Sciences and Technology in Maharaja Ranjit Singh Punjab Technical University, Batinda. As what you have know about what is quality, quality cannot be tested into the product, but it has to be built in design. That is to be analyzed, improve, control, define, and measure. You have to analyze what you have to achieve, what you have to improve in the process of quality, and each of the definitions of each of the measures of quality, and then you have to analyze and build a better product. How can you opportunity to opportunities to accomplish a good quality? This is the main this is the process through which we can get a good quality by plant. Quality is achieved by a plant, not through a chance. There is a process where we can do a process through predetermined. So can we achieve results. What we understand about quality by design? Quality by design is a systematic approach to development that begins predefined objective and emphasizes products and process understandings and process control based on sound science and quality risk management. That is what IC says. That is what we understand about the quality by design. Quality by design starts with a predefined objectives and emphasizes product and process management. Basically, how we want to develop, what are all the processes involved in this developing particular product, controlling all those processes together to achieve a product. This is all known as a quality by design. So, what is the principles of quality by designs? Risk and knowledge-based decision, continuous improvement, capable process making, process development. These all are the principles of QBD, what we understand. And what is you see now in QBD we have to understand what is the risk assessment. We have to do risk assessment through different methods. So we done risk assessment through Ishikawa fishbone diagram. What is man, environment, raw material, method of preparation, target production. These all are the quality attributes of the final products. So we go through the Ishikawa fishbone diagram by assessing all the risk involved in the quality in the formulation of the product. Various aspects that involve in quality by design are critical process parameters, critical quality attributes, and critical material attributes. What is the various critical process? It means that once what we are going to formulate, there are various processes that are involved during the formulation, such as the formulating effervescent granules. There are many processes such as mixing, drying of the products, weighing, temperature measurement. These all are the parameters, all of the process that should be critical in achieving our final goals, that is the effervescent granules. So these are the critical process parameters. What is understand by critical quality attributes? Quality attributes are the final objectives, such as how we characterize the finally our granules, that is known as critical attributes. And material attributes are the major materials or critical materials that are involved for achieving our final product, that is our effervescent granules. These are the critical material attributes for quality by designs. How to QT quality target product to critical quality attribute. These are the methods, so that how we can achieve our quality target product profile to our critical quality attributes, that is the safety and efficacy, identity, potency, purity, others. And there are potential quality attributes for appearance, assay, crystallinity, CU, degradations, disintegration, dissolution, microbiology, stability. All undergoes critical assessment. Then these leads to the formation of critical quality attributes. How a quality attributes called critical? Quality attributes are not all the quality attributes are not critical. Some of the critical attributes are critical. Some of they are not critical. So how we determine our impact to safety and efficacy. Quality impact our final quality, then it is the quality attributes. It's critical. If quality attributes is not going to affect our final product, then it is known as it's not a critical quality. Then how process parameters become a critical process parameter. Critical same in the thinking. If any process involved during the formulation of a dosage form is critical, such as if we are going to formulate granules, then the mixing of all the ingredients are very critical process, very critical is in this. So mixing is a critical process parameter. But other than that, other parameters such as drawing, such as weighing is also very critical, but not very also very critical method. So if any process affect our final formulation, final formulation, final quality of product, then the parameters are critical. Then critical. After that, one of the different ICH guidelines that are involved in our QBDs are international formulation guidelines. Also make strong use of strong QBD in different industrial development of dosage forms, such as ICH Q8, Q9, and Q10 guidelines are involved in ICH Q8, Q9, and Q10 guidelines are seriously involved for the development of dosage forms. Then these are all the steps that involve in quality steps and steps of quality by designs. It is defining UTPP, CQA, prioritizing TPP, and defining QTPP. Again, what is first step of quality by design? How we define our quality target product profile, attributes, critical process parameters. Before starting any process, we have to define that. Then after, we have to prioritize which of them is various important parameters, various important critical parameter and critical quality attributes. Then after that, we have to assess risk assessment and risk management. We have to go through risk assessment and risk management. Then we have, after risk assessment, we have to go through DOE based analysis and experimentation. DOE means design of experiment based analysis. We use particular software. There are various software that use, such as design experts, mini tabs. There are various software for this reason. Then again, then after we have to model validation and defining design space, and then after pilot plant scale up. These are all of the process that we are involved with quality of design. The principle, that is the equation of risk to quality, should be based on scientific knowledge and ultimately leading to the protection of patient. And the level of effort, formality, and documentation of quality risk management process should become commensurate with the level of risk. The method from which ICH Q9 guidelines that says about quality risk management, how we undergo a quality risk management during a dosage form development. First three steps: process development, control, strategy development, and continual step in the quality risk management. Then after risk assessment matrix, risk assessment matrix are those in which we define which of them is high risk, which of them is medium risk, and which of them is low risk. We analyze all those critical attributes and we put down in a table such like that. So red one goes for the high, low one goes for green, and medium one goes for yellow. These are the methods through which we analyze the risk. Then after we analyze the design space, we know that what is design space? Design space is the space in which we perform our experimental activities to achieve our goal of quality product. And it is the structure of 2D based development, that is the defined parameter, design development, impact, identification, and monitor. And for formulation, in this formulation, we are going for discuss about effervescent granules. How an effervescent granule, these are all are the parameters that we go. All are the parameters that it goes for. For different critical quality attributes of effervescent granules, we have to define which of them are physical attributes, which of them is particle size, bulk and tap densities, flow properties, impurities, effervescent time, effervescent sedation time, and so these are all critical attributes that we define for our effervescent granules. E.g., here different for effervescent granules. So here we identify all the critical attributes, and it is critical quality attributes. And yes or no, we in a table we define ki this is the parameter is a critical quality attributes or not. We have a justification for that. We go for that. And then again, this is the target product profile. That the justification for that, we identify all the target product profiles, and how we formulate our effervescent granules. We formulate our effervescent granules through granulation methods, basically in the citric acid, tartric acid, sodium bicarbonate, sodium citrate, dark lactose, in a wet granulation method using forming a dam, then passing through passing through a sieve number 20, then we got drying that granules through in 30 minutes at 60 degrees Celsius. We got our effervescent granules. Here, why we choose an effervescent granules as a product? Here we choose effervescent granules as a product because many effervescent granules can be formed in many flavors. That is for mint cola, orange. It can be formed in any flavor and any taste. So it is a very acceptable dosage form for children's, and also for children who are not very willingly to take medicines in the form of capsules, in the forms of tablets. So those children which are not very willing to take the medicines in that form, in the effervescent granules we can deliver that therapeutic range of formulations through these type of dosage form. This is a novel type of dosage form that we are going to develop, and novel type of dosage form that we are going to develop, so that we can replace our conventional dosage forms through these. And after that, what this is the known as formulation by design. Now what is formulation by design? When we use quality by design in the area of drug formulation or in the pharmaceutical field, then it is known as that area of quality by design is known as formulation by design. It is the same process, identification of factors, CQA, CPP, and then again what do they this is design. We use design of expert software through which our three factors has been selected.
Hello, Dr. Shubnaman. Sorry for the interruption. Your
Yes,
screen has been closed. We are unable to see your PPT. No, not yet.
One minute.
Please share your screen once again. No.
Is it visible, ma'am?
No, not yet. Open your GoToMeetings app and click on the screen button.
Yes.
Yeah, it is visible now. You can continue with your presentation.
Yes, ma'am.
Again, it was
So what
unshared.
we
Kindly.
go through.
Hello, Dr. Shubnaman. Again, it was unshared again.
Very tired. It's visible, ma'am?
No, not yet. Hello, Dr. Shibnaman, are you there? Okay, everyone, let's wait for a few more minutes for Dr. Shibnaman.
Sorry for interruption. There is quite a network issue here.
Kindly share your screen once again.
Yes, ma'am. It's visible. Will be visible now.
Yeah, it is perfect. You can continue with your presentation.
Fuck. Yes. So why would we choose our effervescent granules as our dosage form? Because the children are very much fond about the drinks like colas or orange flavors or Mirinda flavors. So nowadays we are thinking that we can make a dose or therapeutic doses from so that they are more and more attractive towards the children. So we go through our effervescent granules. We formulate an paracetamol effervescent granule through this method of wet granulation method, and that this is formulations by design. What is formulation by design? I previously explained that when we use the formulations quality in the field of pharmaceutical sciences or field of pharmaceutical sciences or pharmaceutical product development, these quality by design are known as formulation by design, and all the character are same in formulation design. And now it is what is optimization in this. We select three factors, that is the amount of tartaric acid, amount of bicarbonate, and amount of PVP, that is the binder. As the three important factors, critical factors, we go through design software and selected responses as effervescence times, Hausner ratios, and angle of repose. These are all are the three responses we have selected and three factors that we have selected. We go in the design expert software and we generate our 2D and 3D plots, which will help us in determining which is critical, what is the critical amount for getting our quality products. And then after that we go through design space determination. But in design space determination, that is all the gray plot that is we are seeing here is where we can go out with our experimental tools, but we cannot get we cannot get our desired output. But when we go in this yellow region, we can get our desired output, what we want to achieve with our final product. So this is known as determination of design space. Then after we go through characterization of our developed effervescent granules, that is key, when we see that key in bulk density is in density, tap density, Hausner ratio, Kar's compressibility index, angle of repose, effervescence time, effervescence cessation times, and also we go for test in very limited area. We go for that all the process, all the characters are in limit, and we can go further testings. And we also go for accelerated stability study of developed model in 3-month time periods, and that our results is quite exciting, and we can go for this formulation in industrial or industrial level or as a new product for this. So with this, what we have learned that we can identify CMA, CPP, and CQN in every processor's life. That is, we can apply quality for achieving a quality in our life or quality in any product. We can apply quality by design in every aspect of our life, so we can obtain our desired outcome. And in pharmaceutical process, we have to understand the process, quality, and characteristic ingredients everywhere, so that we can achieve our quality products. Thank you so much. Thank you for giving me opportunity for delivering a talk here. Back to host.
Thank you, Dr. Shubhnama, for your presentation. Does anyone having any queries? I hope there are no queries. Once again, thank you, Doctor, for your presence and presentation.
Thank you.
Hello, Doctor. Thank you. Hello, Dr. Uchali. Are you ready to give your presentation now? Uchali. Hello, Dr. Uchali.
Hello, can you hear me?
Yeah, are you ready to give your presentation now? Is the issues resolved?
I try to open
Now
my
your voice is clear now. I'm making you as a presenter. Kindly click on the screen button to share your screen.
Yeah.
Yeah, your screen is visible now. Make it in a presentation mode and continue with the presentation.
Okay. Oh, sorry.
Yes, it is visible now. You can start your presentation.
Okay. Thank you very much. The topic of my talk today is a combination of cytokine-induced killer cells with PD-1 blockade and ALK inhibitor shows substantial intrinsic variability across non-small cell cancer cell lines. What I would like to do in the first part of my talk is discuss non-small cell cancer mutations. Lung cancer has been categorized into two main histological groups, non-small cell carcinoma and small cell carcinoma, accounting for 85% and 15.5% of lung cancer respectively. This pie chart shows non-small cell cancer mutations. Mutations in KRAS and EGFR are two the most common genetic events in non-adenocarcinoma and accounting for 20% and 10% of cases respectively. ALK rearrangements are responsible for 3 to 5% of non-small cell cancer cell lines. Nuclear freeze-fall ALK fusion gene was the first alteration in ALK gene to be discovered in human cancers, corrected by a translocation between chromosome 2 and 5. The resulting fusion gene leads to constitutive activation of ALK and downstream signaling pathway that drive oncogenesis. The red diagram shows the fusion of N-terminal portion of EML4, which contains its basic region HELP and part of WD repeat region to the intracellular region of ALK, containing tyrosine kinase domain. The transmembrane TM, this domain is not present in a final fusion product. Rearrangement of ALK with a variety of different circular proteins have been identified. The most abundant are fusion of ALK-G with portions of EML4 in lung cancer. In addition, genetic fusion of ALK with genes such as KIF5B, TFG, etc. have been discovered. Okay. In this study, we select 3 genetically different non-small cell lung cancer cell lines, that is NCI H2228, harboring EFL4 ALK fusion, variant 3B, and HCC78 various ROS1 rearrangement, and A549 KRAS G12S as a control. It is worth mentioning the complex network EML4 ALK protein constructed using a tandem affinity purification approach followed by mass spectrometry. In addition to identifying important roles for molecular node to interact with ALK, such as the adapter protein GRB2 and SHC1, numerous kinases, phosphatases, and scaffolding proteins were identified that play a critical role in mediating survival of EML4 ALK positive cells. This result might be useful for the identification of potential targets of ALK combination therapy. For instance, EML4 ALK protein interacted with CAS protein might involve in cell apoptosis. Additionally, let's, for example, for this protein, CDC37, it's a kind of cell division cycle control protein. It is thought to play a critical role in directing HSP90 to its target kinases. Although in previous study, EML4 ALK protein can upregulate expression of PD-L1 in non-small cell cancers, but there's little known about the relationship of EML4 ALK protein and immune checkpoint. Next, I will introduce the drug crizotinib. In 2007, EML4 ALK fusion gene discovered in non-small cell cancer patients. After 4 years, crizotinib approved by FDA as the first generation ALK inhibitor. Actually, crizotinib is a tyrosine kinase inhibitor. It works by blocking ALK. Additionally, ROS1 rearrangement and non-small cell cancer is also sensitive to crizotinib. It is indicated as the first-line treatment in both Europe and the USA for non-small cell cancer patients with ALK or ROS1 translocation identified before first-line chemotherapy. Next, I I would like to discuss cytokine-induced killer cells, namely CK cells. Despite T cell dominate the immune cell composition in non-small cell lung cancer tumors, the function of CD4 positive and CD8 positive T lymphocyte was dysregulated with a decreasing of intergamma production. In this particular scenario, the infusion of alternative lymphocyte treatment, for instance, cytokine-induced killer cells may help to reshape the therapeutic paradigm. CK cells are heterogeneous in vitro expanded T lymphocyte with a natural killer and T phenotype generated by ex vivo incubation with human PBMC or cord blood mononuclear cells. The transfusion of CK cells after in vitro expression in cancer patients has already been tested in more than 80 reported clinical trials. First, I would like to introduce what are CK cells. CK cells express activating natural killer receptors, including NKG2D, and DNA-M1, and low expression of NKP30. Cell signaling not through TCR CD3, but also through NKG2D, DNA-M1, and NKP30. Let's to activate CK cells, resulting granular exocytosis, cytokine secretion, and cytotoxicity. An antibody blocking experiment show that LFA1, let's say this LFA1 and DNA-M1 leads to significant reduction of both CTL and NK cell function, whereas blocking NKG2D and NKP30 only inhibit NK-like cytotoxicity. Additionally, it has been verified after expansion, CK cells still retain TCR-mediated antigen specificity. Another pathway might involve in Fas and Fas ligand axis. Fas ligand express on activated T cells and natural killer cells poses another way to kill cancer cells by recruiting of Fas associated protein with DD and activating Casper's pathways. It has been found that CK cells are resistant to apoptosis by oppressing anti-apoptotic genes and can produce Fas ligand to induce Fas-dependent apoptosis of sensitive tumor cells. In addition, some therapeutic antibodies such as anti-CD20 antibody rituximab enhance CK cells cytotoxicity via antibody-dependent cellular cytotoxicity, namely ADCC. Once the Fc receptor binds to Fc region of antibody, the NK cell release cytotoxic factors that cause the death of target cell. Next, I will introduce PD-1, PD-L1 signaling axis in cancer cells. When PD-L1 binds to PD-1 expression on the surface T cells, become inactivated through the recruitment of SHP1, SHP2, which subsequently inhibits TCR and CD28 co-stimulatory signaling by preventing the phosphorylation of CAP70 and PI3K, leading to T cell mortality and proptosis. In Figure B, monoclonal antibodies target PD-1, PD-L1 signaling axis has been developed to restore immune-mediated eradication of tumor cells. Next, I will introduce the clinical trials of CAR cells in lung cancer. There's some clinical trials of DC-6 in combination chemotherapy for advanced non-small cancer cell lines. Advanced non-small cell cancer has shown very limited success. To improve the efficiency of such therapies, a number of paradigms has been discussed, including a combination of PD-1 inhibitor with TKI in advanced lung cancer patients. Considering this, we aim to understand whether pre-treated nivolumab CAR cells before transfusion can enhance the anti-tumor immune response to non-small cell cancer cell lines and different concentration of crizotinib in vitro. To achieve this, we use 3 non-small cell cancer cell lines, as I mentioned before, and utilize multiple techniques, including flow cytometry, cell variability as ELISA, RT-PCR, and Western blot. Here I present my data. We have previously reported that CAR cells are composed of 3 main subsets of subsets, that is CD3 positive, CD8 double positive, CD3, CD4 double positive, and CD3, CD56 double positive on day 14. Therefore, we first determine the phenotype of PD-1 and PD-L1 CAR cells primary on day 0 and day 14 for these 3 CAR subsets. The analysis shows that the percentage is the analysis show that the percentage PD-1 positive, CD3 positive, CD3 positive, CD4 positive CAR cells increase significantly after 14 days expansion compared to day 0. However, no significant difference in other subsets. Meanwhile, the proportion of PD-L1 and CAR cells will also increase significantly among the 3 subsets on day 14. To mention, we confirm that non-small cell or 3 non-small cell cancer cell lines express PD-L1, but at different levels. Next, we examine the association between CAR cells activation and PD-1 surface expression in the presence of nivolumab and found that PD-1 expression on the surface of CD3 positive CAR cells decreased significantly from 4% to 0.4% after 48 hours treatment with nivolumab compared to IgG4 isotype control. Also, the assessment of mean fluorescence intensity, that is MFI, of surface expression of potential immune associated markers, including PD-L1, PD-L2, CD28, CDR4, GITR, and CD134 on CD3 positive CIK cells showed no alterations. When counting the absolute number of PD-1 positive CIK cells using counting beads, there was also a notable decrease in both PD-1 positive and CD3 positive CIK cells. All PD-1 positive NK T CIK cells after the nivolumab treatment. However, PD-1 mRNA levels were not significantly changed by nivolumab. This finding suggests that nivolumab blocks PD-1 on the surface CIK cells. Interestingly, nivolumab caused a significant increase in the level of CD4 positive, CD25 positive CIK cells expression, FOXP3. Sorry. Considering the nivolumab might reduce tumor cell viability, we performed CCK8 assay and found that the combined treatment of nivolumab with crizotinib significantly impaired the viability of NCI H2228 cell lines. Unfortunately, no such change was observed in other cell lines as well as CIK cells. Yes. Suggestion that this effect is cell line specific. To assess absolute quantification of dead tumor cells, let me directly reflect the cytotoxic activity of CIK cells. We labeled the non-small cell cancer cells with a specific dye reagent, let's call it CFSE, and culture with nivolumab pretreated CIK cells and effect target ratio of 10:1 in the percent of crizotinib. The absolute number of dead NCI H2228 cells were found to be significantly elevated after combining different concentration of crizotinib. Here again, two other cell lines, HCC7H and A549, as show weaker sensitivity to CIK cells cytotoxicity when combined with crizotinib and nivolumab. To investigate the potential variations in immune markers and cytokine primarily associated with cytotoxicity of CIK cells, we subsequently examined infrared gamma expansion by ELISA and intracellular expression of GRAZB by flow cytometry. Previously, CIK cells were reported to express high percentage of GRAZB, that together with another potent porphyrin mediate apoptosis in target cells. Our results show that CIK cells respond to a combination of PD-1 blockade and crizotinib against NCI H2228 with a strong secretion of interferon gamma and a significant increase in GRAZB. However, no significant change was observed in the HCC7H and A549 cell lines. This result. Considering that cell surface fast ligand expression is specific to the immune system, we next investigate fast ligand surface expression on the CIK cells. The flow cytometry analysis show fast ligand is low level surface expression, counting for less than 5%. Then we further verified using Western blot method. Importantly, nivolumab combined with 1,000 nanomolar crizotinib resulting significant upregulation of intrinsic Fas ligand expression compared to the untreated CIK cells control. In summary, we have verified that PD-1 surface expression of CIK cells decrease. Dramatically after treatment with nivolumab, and it had no effect on PD-1 mRNA expression in CIK cells. Nevertheless, nivolumab was found to bind to T lymphocytes for more than 20 weeks in non-small cell lung cancer patients, even after stopping treatment, suggesting a sustained therapeutic effect. Secondly, blocking of PD-1, PD-L1 pathway by nivolumab increased cytotoxicity of CIK cells targeted to NCI H2228 rather than HCC78 and A54 cell line. Finally, the evidence obtained from multiple markers in our study, such as increased production of lymphogamma from CIK cells, activation of Fas ligand in CIK cells, and increased intracellular expression as graphically of CD3, CD56 positive CIK cells, suggesting the combination of nivolumab and crizotinib accelerates the release of immune-related effector molecules. In conclusion, CIK cells in combination with crizotinib and nivolumab can enhance the anti-tumor immune response. Studies suggest that genetic background play a significant role, and the combination therapy should be optimized by considering genetic background and immune response. Thank you very much. Okay. Thank you.
Thank you, Dr. Utelli, for your presentation. Does anyone having any queries? I hope there are no queries. Once again, thank you, Dr. Utelli, for your presence and presentation. Okay, now by this, we have come to an end of day one webinar. I would like to thank all the attendees and participants of today's webinar. Hope you enjoyed the webinar and gained knowledge from the sessions, and request you to attend tomorrow's sessions too. If you have any queries, you can mail us. And thank you. Please join tomorrow's webinar too.